Greetings. Welcome. We'll get started here in just about 1 minute. We'll give folks a bit of a chance to get in. But welcome, welcome. I see we've got folks joining us. All right, we'll get started. Well, welcome to the 2024 NAISMA Annual Biocontrol Summit. My name is Christy Trifone-Millhouse, and I'm your NAISMA Executive Director. A few house cleaning items before we begin. I'd like to invite you all to share with us in the chat where you're joining from, and take note of anyone you might know and send along a hello. Additionally, you will see the reaction button at the bottom of your screen. Feel free to use these after each presentation. The Q&A box is where we ask you to put all questions. If you add them to the chat, we will not be able to answer them. So please add them to Q&A. And finally, this summit will be available after today's event on the NAISMA website. So go to naisma.org for more info. Our theme for this year's event is Aquatic Weed Biocontrol, Project Progress, and Innovative Tools. For many, this will be the first time you learn about biological control efforts. For others, you're working in this space every day to expand and improve this management tool. No matter what your experience base is, we hope that you will learn something new during today's event. Before we begin, I'd like to share with you a bit about NAISMA. We're a 501c3 organization with a mission to empower invasive species management in North America. Over 30 years ago, NAISMA's founders formed this organization that started as a grassroots movement and grew into an international collaboration focused on supporting invasive species management. NAISMA continues to grow by leaps and bounds every year. We currently have over 800 members from all over North America and beyond. We're active on many social media platforms, and I encourage you to follow us. NAISMA creates bridges across geographic divides and jurisdictions. Our programming focuses on our 3 pillars of support: the connections, the voice, and the tools. NAISMA is the organization that provides a collective voice for our members and invasive species community as a whole on a national and international scale. There are over 800 registered for our summit today. Many of you are not members. I'd like to encourage you all who have not become members to do so. Your membership helps NAISMA to continue to provide these trainings from experts in the field to all managers across North America. In our current membership structure, we have options for individuals and organizations. We will be expanding these offerings over the next year, so please stay tuned. All right, now I'd like to recognize the NAISMA Classical Biocontrol Committee members who work hard to bring biocontrol education to all of you. They have helped create weed and biocontrol fact sheets for 46 systems and a Biocontrol 101 Invasives U course. All are available on the NAISMA website. A special thank you to those members highlighted in blue who are responsible for organizing this year's summit, particularly the co-leads, Melissa Maggio and Jennifer Andrews, along with our NAISMA staff member, Jody Hogan. Finally, I'd like to thank the USDA Forest Service, who generously provided funding for this summit and made it possible to offer this as a free event for everyone. It's now my pleasure to pass the mic over to Melissa Maggio, the director of the Montana Biocontrol Project, who will be moderating today's event. Thank you so much for joining us. Melissa?
Thanks, Christy. And to all the attendees, welcome to our annual Weed Biocontrol Summit. Thanks for joining us. As Christy said, my name is Melissa Maggio, and I am a co-chair of NAISMA's Biocontrol Summit Planning Subcommittee. We are joined today by Jennifer Andrews, the other Summit Planning co-chair, who will be managing the question and answer box and selecting questions to ask our speakers during scheduled question and answer periods. This year, the summit will highlight the role weed biocontrol can play in managing invasive aquatic plant species throughout North America. Our invited aquatic weed management and biocontrol experts will share updates in biocontrol development and innovative tools for improving integrated weed management. The NAISMA Classical Biocontrol Committee wants to continue to strengthen and expand our biocontrol community, and your input is valuable. We will be sending out a post-summit survey and would appreciate feedback on content, future events, improvements, and community growth opportunities. You are also welcome to contact any of us directly. A little bit of housekeeping before we get started. During our scheduled during scheduled periods, we will have a formal question and answer. Please type any questions you have for our presenters into the Q&A box, not the chat box. And they will either be answered live or answered in that box. If you're asking a question of a particular speaker, please make sure to address them directly. Also, if you have any technical issues, please put those in the chat box, and Jody will do her best to help you out with those. You can also email her directly at assistant@naisma.org, and I believe Jody put that information in the chat box. Jody will also be putting a link to the agenda in the chat box in case you don't have that on hand. Okay, so if we can stop sharing the agenda, and we will welcome our first speaker to begin sharing his presentation. And I am delighted to introduce our first speaker, Dr. Al Cofrancesco. Will kick off our summit by providing an overview of the last 60 years of aquatic weed biocontrol. Al, are you there?
Hello, do you hear me?
I can hear you, yes.
All right, let's see if I can get the share screen. Do you see it?
I'm seeing your note page, so you can just go ahead and swap. Hopefully that will work.
Okay.
It looks great.
Okay, great. Thank you very much. Sorry, I'm not as technically capable or savvy as other people have been, but I want to thank everyone for allowing me to make this presentation today. When I got into this study, starting to look at it, it's brought up a lot of memories. I've worked in this field for 45 years, and so it was a joy to go back and start looking at activities. Actually, it was also a problem. There was a lot of information, and I hated to leave things out. I hated to look at what was done and not make mention of it. But based upon that, I really think I'd like to write a history of the aquatic weed biocontrol because of the plethora of information out there. So we're going to hit a few topics today, and we're not going to cover it all. It's impossible to do that. But basic foundation for those people who aren't as familiar with biocontrol, what we look at here is the fact that when a plant comes into a new territory, it could become invasive, and it goes from a non-problem level to a problem level, and it stays and it oscillates in that area. What we try to do is take and find, say, insect biocontrol agents or some host-specific agent that will impact that plant, reduce it and stress it, and allow it to go back to non-problematic levels. This has been seen in biocontrol for, say, terrestrial systems. In the late 1940s, 50s, you saw that the release of chrysomelid on klamath weed had a devastating impact on vegetation and was sort of the silver bullet for the activities. So people started to feel like, well, maybe this is the way or the approaches that we should use for aquatic plant management. Now, over the course of time, this is just a list of some of the biocontrol plants that were examined and used to work on biocontrol for. Right now, I can't go through this whole list and discuss each one of them. What I've done is I've selected about 4 of the plant species. We're going to talk about those and see how research was conducted on them and what the results were. And then also I want to talk about what we should do in the future based upon the information that we've gotten from the past. Okay, to start it off, alligator weed, Alternanthera phylloxeroides, in 1963 infested about 97,000 acres of the southeastern United States, causing significant problems to the Corps of Engineers. I, being a member of the Corps of Engineers, but I wasn't around in 65 to do it, this is a problem for us. And why is it a problem for the Corps of Engineers? Well, the Corps has waterway systems. They act as a conduit for the movement of aquatic invasive species. We maintain over 12,000 miles of navigable inland waterways. Reservoirs, dams, etc., all are impacted by this vegetation and growth. In the 1960s, the way we were treating alligator weed at the time frame was to use a herbicide spray. Now, people don't realize this. Alligator weed actually outcompeted a plant called water hyacinth. It overgrew the plants, became very established, and was causing a lot of problems. In addition, alligator weed was shown to be resistant to the herbicides that were in use at that time frame. Herbicides would burn it back to the water surface, but couldn't translocate below the water surface. So the Corps of Engineers went ahead and got and collaborated with, or funded really, the USDA in 1960 to develop biological controls, or to look for biological controls in South America to control or manage alligator weed. Now, I like to point out here, in going back to the literature, these researchers here were key, foundational people who took information from the terrestrial systems and applied it to the aquatic systems, worked very diligently. And I happen to say that I've known most of these individuals. And actually, David Perkins, who's still alive, I contacted him when I heard that I was going to try to get this paper together and talk to him for about an hour. David's insight into what they did in the laboratory was outstanding. And I probably could give a whole hour lecture on just what David and I talked about. But the main thing I really want to point out is these people were very diligent, hardworking individuals, and they ended up using the funding from the Corps from the 1960s on that we funded on an annual basis to develop the South American Biological Control Laboratory for the USDA. So this is how this laboratory was founded, and it did work not only on aquatics, but on some terrestrials also. Okay, alligator weed insects that were looked at and found, initially, Agasicles hygrophila was the first agent that was released in the United States. The alligator weed flea beetle. Think about this, though. Funding was established in 1960, but the first agent came out in 64. That's, to me, almost like a record in how fast the biocontrol agent has been developed. In addition, they found Aminothrips andersoni, which was released in 67, and also a alligator weed stem borer moth. I still call it Votium alii after George Vogt, but anyway, that was released in 1971. And again, the collaborative research efforts. The USDA would do the overseas research and the quarantine work. The Corps would help with the release and establishment work and the distributions. I want to point out 1971 and remember that date. So later on, we'll talk about it. But what we were seeing is this kind of a devastating impact. I like this slide because it shows a number of things. It shows how host-specific the biocontrol agents were. They devastated the alligator weed in this site. They just knocked it back in a very rapid period of time. Also, you can see how alligator weed was trying to outcompete water hyacinth here. So we show the host specificity of the organisms also. They don't attack anything else. They do attack the target plant. We saw this significantly throughout the United States. In South Florida, in Alabama, early in the season up there in the Ortega River, that's the initial site that was thick with flea beetles. And by the end of the season, it was completely removed. So the impact that the biocontrol agents were having was very rapid, same that they saw with the Klamath weed. So they thought, hey, we have a silver bullet. This is the great process. We need to continue this process to be able to go forward. And this is a schematic of sort of what activities are needed. Now, early on, we were doing more overseas research, quarantine, and release and establishment. We really didn't do as much evaluation, tech transfer, or foundational research. That came later on. But this is the gauntlet. This is the system that's being used, that's in place today to bring biocontrol agents in. So now, hey, we had a success. What's the next plant we tackle? Water hyacinth. Water hyacinth biocontrol was looked at, again, from South America. These agents, this plant was a tremendous problem for the Corps. The Corps was looking at very archaic mechanical harvesting methods or chemical control methods to try to stymie the growth or the distribution of this vegetation. So in South America, those researchers that we talked about, they turned their sights, and at the same time they were working on alligator weed, they also worked on water hyacinth. Nicotina icorni, the model water hyacinth weed, was first released in '72. So the last alligator weed the last alligator weed agent was '71. This is '72. They had a production line going here. They were moving things out. Nicotina brookie, the next agent, came out in '74, and then the moth came out in '77. Megamelis is a newer insect that's put out there because of the impacts that that the insects have had. It hasn't been as devastating as other things. They wanted to look at additional agents. Now, I want to point out the two weevil species are slower growing, and their life cycle is completely tied to the plant. And this has a implication later on. Well, when we looked at some data that we had from the Louisiana Department of Wildlife and Fisheries, we could see that before the releases of weevils in Louisiana was in 1976. But the plant has the capability, if you look at the diagram, of growing about 1 million acres in a growing season. The winters would knock it back to about 200,000 acres, and at the peak of the growing season, we'd get up to 1.2 million acres. But even with the release of the insects in masses out there, we kept seeing that the water hyacinth would keep growing back. And so it wasn't the silver bullet that happened with alligator weed. So people were starting to get discouraged. But if you look at what happens over a long period of time, you can see that eventually the degradation by the insects, the trial feeding on it, actually caused a complete collapse of the water hyacinth, and it continues and is maintained today. This is a 38-year period of time that we're looking at right here. But in areas where water hyacinth was left untouched, where insects could build up, where herbicides weren't used to spray and kill off the plants, because when that happens, it takes out also the adults, takes out the larvae, the pupae, and the insects cannot rapidly reproduce. So the water hyacinths come back rapidly. If you let it stay, like we did here in Texas on the Trinity River, you can see that we saw a rapid decline over about a 3-year period in this open area here on the bottom. While this was all going on, Salvinia molesta was also being looked at, but this was being looked at by the Australians. It wasn't a problem in the United States at this time frame. Back in 1980 or so, they were releasing the Salvinia weevils, sort of back at Salvini, and this is work done by Ken Harley, Wendy Forno, and saw a dramatic release, a dramatic impact. Within 14 months, 200 hectares of Salvinia was under control in the lake. This small weevil really devastated the plant. This plant was hard to control because of the hydrophobic hairs. So when herbicides were sprayed on it, they really had to put a surfactant on it. So it didn't work very well. But they had such vast amounts of Salvinia that they used this biocontrol agent, which worked very rapidly. A slide I got from Mick Julian, where they worked in Papua New Guinea on the Sepik River here in 1982. This was the infestation you saw. Then about 2 years later, this is the site, completely open water. So basically, we have another agent now, another agent that's very rapid on a target plant. So we've had one. Alligator weed insects seem to work rapidly. Water hyacinth take a longer time to impact the plant. Salvinia, molesta, insects seem to work rapidly. Next plant that was brought up was Hydrilla verticillata. Okay, we need a biocontrol agent for this plant. Now, Hydrilla is not native to South America. We had the laboratory established down in South America, but we have to look where overseas do we look for biocontrol agents of Hydrilla. Where's the best location? Where is Hydrilla native to? In getting with the botanist and trying to determine this, we honed it down to only 3 continents: Asia, Africa, or Australia. I mean, this is a very difficult situation to try to look for a biocontrol agent on the land masses, which are probably 2/3 of the world's land masses. You have tools today which are better to assist you, being able to genetically examine plants and being able to determine which species or which genetic genotypes you're dealing with in, say, the United States, so that you can go to regions of the world and look where those particular genotypes are and be able to look for biocontrol agents, rather than just look for a plant that looks morphologically like the plant we have, but maybe the agents aren't really agents that would feed on it. Anyway, work was done with USDA in collaboration with CSIRO and the Corps of Engineers. They found a number of agents. Bagus affinis was found in India, and it works excellently on the tubers. The problem is it only feeds on the tubers in the dry season. When water is off the site, it would find tubers and devastate the tubers, but we don't find that situation in the United States. So they really are not effective as a biocontrol. The Bagus hydrilla, this agent really was found and feeds on plants, very host specific, but doesn't do a lot of damage, and only is really found up more in the windrows when plants build up on the banks. These eggs were released in the United States. 2 other agents, Bagus, Hydrilla pakistani and Hydrilla balchunicide, were both released in the United States and do damage. And we looked at how damaging they are. They take out the mesophilic tissue in the plant, reducing the photosynthetic capability. You can see here how the plants are very limp and deteriorating. So we were hoping that this plant would either get secondary infections, or the insects would be able to reduce the ability of the plant to grow. And we were hoping to monitor this and see. Now, some sites, like the site here in Texas at our Louisville facility, we saw extensive damage and a dropout, but we couldn't really prove that it was the insects themselves that did it. There's a density of them and the impact, but we couldn't predict it at all locations. Maybe it's a long, drawn-out affair like we see with water hyacinth. We have to look at data for 20 or 30 years before we start seeing a natural decline. But we still need to start looking and continue looking for biocontrols. At the same time, I want to point out that we had some researchers being done, Glomsky in Netherlands, showed that, if they just took 1 sprig of hydrilla, 5 centimeters, and planted it and let it grow for 5 weeks, it grew to 266 feet. So this is a plant that had tremendous regenerative capabilities and moved very rapidly. This kind of scenario is seen with all the biocontrols and the plants that we're dealing with in the United States. I could go through the whole list of plants, and it would be more than an hour's worth of time. But what I want to do is turn my attention back to some work that was done with the water hyacinth. Water hyacinth, as we remember from the slide here, we released weevils in 1976, and you could see the impact that was done. But what is the benefits that are derived from it? We need to be able to look at data and show how the management of this invasive weed benefits society, not just how much impact we're having on the target plant. People look at us and ask, why are we doing this? And why is this cost effective? And why am I putting money towards it? We need to be able to answer those questions. So we got with the University of Maryland and worked with them, because we looked at commercial navigation or commercial fishing, flood reduction, and non-use areas for ecosystem services. And we have to start to look at that and ascertain what's happening there. We need data collected. Some of the data we collect, but some of it we don't. How many days are clogged or for flood control structures? How many days need cleaning? What's the cost of cleaning? We have to really start as biocontrol people to also collaborate with economists, environmental economists, and modelers to be able to help to ascertain what's actually happening. It might be a slow, dramatic impact, but we need to be able to showcase what's going on. And then we need to be able to present it in a way that society and the public understands. Okay, for water hyacinth, they impact these 8 issues. But what data did we collect? When we started the studies, we never looked at recreational fishing, recreational hunting, etc. We never had data and information. But getting with the economists, etc., and people who understand how to look at these, we were able to try to put together an approach. We wanted to look at how to develop the intrinsic values that are there, and then look at the economic performance, the changes and benefits that are derived, and then how much it costs to get that benefit level. If we look just at one characteristic, if water hyacinth was left unchecked and it hit all open water in the state of Louisiana, we modeled it and saw how many acres would actually be infested. This is a tremendous help for us to be able to showcase what is going on. Okay, in looking at the benefits derived, we were only able really to ascertain maybe 5 different areas that we could get good data on. But we got data over a 38-year period of time. And in calculating it, we saw that to a total of $4.2 billion in ecosystem service benefits resulted from us maintaining water hyacinth at 100,000 acre, 200,000 acre level in the state of Louisiana. We turned our attention next to how much did it cost us over those 38 years. And we looked at how much we spent on biocontrol agents, how much we spent on herbicide control, because we really couldn't tease out the herbicide applications. There was an active spray periods going on, and in high use areas, water hyacinth was sprayed every year just to remove it. But the insects were not having the impact because the life cycle of the insect was disrupted. But in the backwater areas, that's where the water hyacinth insects really did their greatest damage and reduced the acreages. But for a total over the 38 years that we spent was $124 million. So if we look at this, then the study indicated that biocontrol coverage in Louisiana would be on the order of 45% higher in the spring and 73% higher in the fall if we hadn't used the biocontrol agents. Again, we mentioned the cost, $124 million we spent on it, but the benefits generated were $4.2 billion in calculation, a benefit to cost ratio of 34-1. It's like that the benefits derived, we were understanding that we needed to be able to obtain other data like commercial navigation or flood damage reduction. These are areas we weren't able to get good values for. So really, I think that the benefits generated would be much, much higher if we were able to incorporate those into the system. But what it did prove to us was that even though we couldn't separate out the herbicides from a demonstration and the synergies that maybe occurred between them, our study suggested that the impact was there. And it also just suggests that information doesn't have to be perfect to conduct a complete environmental or economic analysis. We get orders of magnitude of benefits that are derived, and we need to be able to showcase this to the population. So in conclusion, what I want to say is it's been a very successful run with aquatic plant biocontrols, but we need to continue to monitor the impact of the biocontrol agents. The impact may take time to occur. We need also now in the onset to consider climate change and the needs to be considered how and what to monitor for a long-range understanding of what's going on. We need to utilize the new technologies on the table, genetic analysis and environmental modeling, to help us assess what the biocontrol research is being conducted and how to move forward in those areas. We need to consider analyzing ecosystem goods and services and understanding the benefits derived by biocontrol management. We need to be able to present information to the public in terms they understand. These are areas, I think, that we haven't really addressed very well. The public doesn't understand the good that we're doing, and the services that are being rendered, we just accept as anything that would occur. They don't realize that water hyacinth at times we're growing 1 meter tall, and we're over 1.2 million acres in the state of Louisiana. I thank you, and if there are any questions, I'm more than happy to answer them. Thank
you so much, Al. That was great. It doesn't look like we really have a lot of time for questions right now, but we will have a question and answer period at 12:15 Central Time. So at that point, there might be some questions that Jen asks of you.
Thank you.
Thank you so much. So you can go ahead and stop sharing if you would. And then our next speaker can go ahead and bring up her slides.
Can I replace current share?
Oh,
there we go.
Okay, there we go. Okay, so our next speaker is Julie Coetzee with the South African Institute of Aquatic Biodiversity. Welcome, Julie.
Thank you. Am I sharing correctly?
At the moment, oh, it's your notes pages.
Oh, not again.
Yeah.
Right. Is that
Never what you
correct?
expect. That looks perfect. Thank you.
Great. Well, I think Al and I had a really great rehearsal in that his talk ended with hippos in water weeds, and I've started with hippos in water weeds. So completely coincidental. So I work for the South African Institute for Aquatic Biodiversity, but I'm also the Deputy Director for the Centre for Biological Control in South Africa, where we have a pretty big program on biological control of aquatic weeds. And today I'm going to be talking about the inundative biological control program that we've implemented to get unbelievable results with water hyacinth biological control over the last couple of years. So I'm obviously right down here at the bottom of Africa, and this huge continent has got a number of terrible floating macrophytes, which I'll introduce: the water hyacinth and giant salvinia. We also have water lettuce as well as parrot's feather. And the latter three are all under really effective biological control, classical biological control, but we still really struggle with water hyacinth. And if we have a look at the water hyacinth problem in Africa, and really emerged as a huge problem in the 1990s, this is what Lake Victoria looked like in Kenya. So it's Africa's largest lake. It's a huge source of income for many people whose livelihoods depend on access to this water. And if you can think of what this devastating mat of water hyacinth would have done to the people living around that area, it was hugely significant. 1998, this is another picture taken from another angle of the yacht club. And in the 90s, biocontrol scientists from the USDA, TED Center, Mick Julian from CSIRO, and as well as Martin Hill from the Agricultural Research Council in South Africa, started working on biological control of water hyacinth in Africa, and in particular on the system. And what they set up was mass rearing stations, where people living around these lakes, and this actually isn't Lake Victoria, this is Lake Malawi in Malawi, but a very similar thing, rearing insects, the two weevils that Al spoke about, the two neocotynas, rearing them on the banks of these huge infestations and allowing people from small fishing villages who had essentially lost their livelihoods, who had lost access to water, lost their transport mechanisms, to take control of this problem on their doorstep. And these are some of the people from these fishing units, and it's blue swimming pools, blue plastic swimming pools that were sent from South Africa to the banks of Lake Victoria, to Lake Malawi. And here people could rear these insects with pretty basic technology and be a part of the solution to an incredible problem. And by 1999, this is what that same picture, the Kasumu Yacht Club, looked like. So similar to what Al was showing, this incredible decline in the coverage of water hyacinth. So on this system, it went from 200,000 hectares, and I don't know what that is in acres, down to less than 2,000 hectares. So phenomenal results. Now, that was just with the 2 neocotinal weevils, which are the mainstay of biological control throughout the tropical areas of the world. In South Africa, we've released more biocontrol agents against water hyacinth than any other country. There's those weevils. We've released a mite, a pathogen, the same moth that Al spoke about, as well as 2 mirids and a grasshopper. And despite these this diversity of control agents and the amount of damage. I mean, here you can see how incredibly damaged these water hyacinth leaves are from the feeding by these insects and the mite and the pathogen. We still end up with really impacted systems. So a huge amount of damage, but very little control. And there are a number of reasons for us, but I'll touch on 2. Unfortunately, we have a tsunami of human waste inundating our rivers and dams. In South Africa, we've got very poor wastewater treatment infrastructure. It's old, it's failing, and virtually every municipality in South Africa is pumping untreated wastewater into our aquatic ecosystems. And we know that eutrophication limits the success of classical biological control. So as fast as these insects are feeding on the plant, this plant is just increasing or growing in as fast as it can. You know, it really isn't being impacted too severely by all the damage from these different agents. And then another aspect, which is very relevant to the US and that we deal with as well, is that we've got these tropical species that have originated or evolved in low altitude warm areas, and they've been imported into, in South Africa's case, high altitude cool areas, cold frosts over winter. We don't really get snow. And so they're physiologically not capable of overwintering. And we had a really great collaboration with Nathan Harms and a whole lot of collaborators, some you might see a bit later, looking at how this climate mismatch, how we could improve this climate mismatch through new innovations in biological control. And so in 2013, we released a new agent, the hopper Megamelus scutularis, that we actually imported via Florida through the USDA. And we completed our host testing and we're able to release it. And now you might be questioning, well, why are we doing this when we are facing the same, it would be facing the same problem of high nutrient waters and cold winter temperatures. So the area that I'm going to be focusing on is where this blue dot is. It's where Johannesburg and Pretoria are, the capitals of South Africa. Very cold winters, these high altitude sites. And there's a whole series of impoundments that are invaded by water hyacinth and that are highly eutrophic, hypertrophic, as well as experiencing these cold winters. And so this is the dam that I'll be talking about in particular. It's an impoundment. It's about a 2,000 hectare dam, and water hyacinth has been present on it since the 1960s. It's been controlled through various methods, mostly chemical control, but that was stopped in 2016 for a whole lot of reasons, mostly political. And that's what the water looks like. So it's receiving all of the effluent, wastewater, untreated wastewater, agricultural runoff from Johannesburg and Pretoria, Johannesburg is the biggest city in South Africa. And so just a hotbed for these blue-green algae, as well as water hyacinth. And in the 1990s, all of the available agents in South Africa had been released on the system. But due to, as Al mentioned, this interference from herbicide and these cold winters, we never really got the control that we would have expected. So in 2018, we started we decided to start on a new type of biocontrol program. So this is what the outline of the dam looks like, and the green is obviously water hyacinth. And on this day, on the 18th of March, this is how damaged the plants were. So in the absence of herbicide, the weevils had built up, the mirid was there, the fungus was there, the mites were there. But this was relying on classical biological control. So in 2019, we embarked on an inundative mass release campaign of Megamelus, where every 2 weeks, we'd release between 6,000 and 10,000 of these megamelis throughout 2019 and into 2020. And the way that we did this was through a mass rearing facility. So we employ people from near the university where I work at, Rhodes University. They come from the local community, and they're all physically disabled, but they're able to rear insects for us. And the sesonke, that word there means we work together. And these guys grow the plants, they rear the insects, they collect the insects, and then we can send the insects off in food containers all around the country. And we really started this at Hartbeespoort. So in 2019, we had 35% cover of this dam, about 700 hectares. And now you must remember, we are in the Southern Hemisphere, so this is the peak of summer. The 3rd of December, that cover had gone down to 20%. And this was quite unexpected because this is the peak growing season for water hyacinth. So when we went to have a look at these plants in January 2020, it looked as though they had been herbicided, but only the water hyacinth was damaged. That other green stuff or other aquatic plants growing on top of it. And when we investigated closely, megamelis had exploded, Neocotina had exploded. And by the end of January, we were down to 5%, less than 100 hectares. So this was phenomenal. This was something we had never expected. July is our winter, and that small green bit was present throughout the winter through COVID. We could monitor it through these satellite images. But by October, we were starting to see the green coming back. And that was because water hyacinth has these incredible seed banks that last approximately, apparently the seeds last for up to 25 years, and they can produce thousands of seeds per plant. So a huge seed bank. And what we were seeing was these seedlings germinating en masse. By the beginning of December, back up to 42%. And, we just thought everything we had done was we'd lost. But we carried on releasing. So there we are, still releasing, but also we set up these satellite rearing stations, again, in the same way that the old white and hairies had done in the 1990s. We did a similar thing around the system where we set up rearing stations with the communities, and we got them to start rearing these insects themselves, megamelis, and it's a very, very fast, it's got a very fast generation time. It lays hundreds of eggs. This is me giving some kids a talk about it during COVID. And once again, these bugs exploded. By the middle of February, we were down to 17%. And the most remarkable explosion of insects occurred. And they moved en masse off the water hyacinth plants because of the food quality was so bad. That's megamelis swarming under a tennis court lights. And that is what the guy who had to clean up the tennis court had to deal with the next morning. So just millions of megamelis that were in search of healthy water hyacinth and food. So by the end of March, we were back down to 7%. And we've done this now for 6 years. So 6 years of community engagement with biocontrol implementation and monitoring of water hyacinth. This is, again, a picture of the dam, and these are where all the stations are. And really what we can see here is that since January 2020, following the introduction of megamelis, every year we get this peak in water hyacinth followed by a peak in megamelis and a crash over winter. And the stats show that it is due to megamelis. And what we really want to show is that by switching from a classical to an augmented biological control program, even though we know water hyacinth is limited by all these confounding factors, inundative mass releases in the absence of herbicide can result in control. It's really important for stakeholders to be involved, and it also offers training and employment. So thank you very much. Escape.
Thank you, Julie. That was great.
Thank you. And I'm just trying to see how to stop sharing.
It might be down at the bottom. You figured it out.
Oh, there we go. Okay. Thank you.
Thank you so much. All right, our next speaker is Melissa Smith with USDA ARS Invasive Plant Laboratory.
Thank you so much. And it's such a pleasure to go after the previous two speakers. Al and Julie have absolutely paved the way for all of us. Okay, let's get this started. So I'm apparently talking about innovations and developments for biological control of water hyacinth, but I want to preface this with there are no new insects. We are all still dealing with the same ones that have been out for well over a decade. So what I'm really going to be talking about is how we're incorporating newer technologies to understand this system better and hopefully get better outcomes in the meantime. So let's talk about water hyacinth for just a second. This beautiful little diminutive flower, which produces both vegetatively and by seed, as we've discovered, has a giant following throughout the world, including in pond and aquaculture, which is how it is still spread around the world today. So, and just a quick search, I found a couple of different videos highlighting how amazing water hyacinth is for your pond. And then you can even go and purchase it for the said pond at any available eBay seller. So if anybody's looking to make a, just kidding, this is a noxious weed, so please don't do this. But it does illustrate the fact that not only is this still a problem with already established populations of water hyacinth, but with water hyacinth, we have new potential invasions happening all the time. And once it gets out, we saw some pretty spectacular pictures of water hyacinth. This is what it does in the system. And once upon a time, our only method of control were these very inexact and non-specific harvesters. Believe it or not, we still use harvesting in certain areas, and there's a big movement to move towards harvesting. But you can imagine that it doesn't just pull in water hyacinth, it pulls in everything else, including invertebrates and vertebrates. So it's a really inexact method. As we developed other chemical methods to control plants, we started to use those. So 2,4-D, glyphosate are both really effective at treating water hyacinth, but they can be toxic to the environment as well as they're non-specific. So the major difference, and I'll hit on this a little bit later when we're talking about more specific chemistries, the biggest difference between either one of these and biological control is that, as Al explained at the beginning of his talk, when we're using biological control, we're using host-specific natural enemies to control our target weed. That means when they are given the opportunity to eat something else or escape, just like those megamelis that Julie showed us, they have to escape because they cannot live on anything else except their target weed. And for water hyacinth, we have a myriad of potential insects. We do not have Acritis tarsus in Florida, but that is used elsewhere. But what we're mostly talking about with effective biological control for water hyacinth are these two guys at the top, the Neocotina weevils, and then the insect we just talked about with Julie, the Megamelis scutularis leafhopper, which has been out since 2010. And these guys are actually pretty effective when left on their own. So the picture on the left is water hyacinth that's been controlled using insecticide to keep insects off, and they do a great job. They reduce biomass by 58%, reduce flowering by 97%, but unfortunately they only reduce coverage by about 17%, and that's where we get into having to integrate other technologies. So for this talk, I'm going to organize it into 4 different sections. One, we're going to talk about increases and advances in monitoring and surveillance, and a lot of that comes out of the Center for Biological Control in South Africa, as well as some really neat public access innovations. Second, we're going to talk about understanding the invasive dynamics, that bottom of the flowchart in Al's talk, talking about foundational research. We need that for water hyacinth because we're still trying to crack this nut. I'm going to talk a little bit about studies that we're doing to develop best management practices using integrated methods. And then finally, I'm just going to make a big appeal to already friendly ears to establish a working group working on water hyacinth. So the first thing I want to introduce you to is this. This is Google Earth Engine, and it was developed a couple of years ago, and it is freely and widely available. And it uses a couple of different Landsat imagery sources here. So satellite imagery that is in the sort of mid scale. So somewhere between 10 and 30 meter resolution. You can plug in your own algorithms and then apply those to the globe, so sort of a geospatial context. And there's all kinds of applications you can use this for, but increasingly people are using it for water hyacinth and to measure water hyacinth, as well as a myriad of other weeds. Just one of the great applications that's been used, again, out of Center for Biological Control and Rhodes University is Dave Kinsler's model, looking at these specific water impoundment areas and then looking at changes in water weeds. So, and it's usually using NDVI, so normalized vegetation indices. And basically that is a proxy for how much green is on the lake. So you can tell the difference between lake and plants growing in the lake. And we can see that over time. Again, a series of different potential satellites you might use, and we can see that the spatial resolution, especially in these Sentinel satellites, is quite good. 5 to 10 meters allows us to tell a lot of information. And that passes over between every 6 and 12 days. So we're getting a huge amount of information that allows us to analyze all of this. And these are publicly accessible. I want to repeat that. Another good picture, so Julie showed this, which is, again, we can calculate the macrophyte cover on those dam impoundments. Here's another one, which associates the decrease in vegetation with the release of biological control, as well as an influx of eutrophic waters. We also deal with that here in South Florida. So again, just wanting to really show the application for this from several authors in several places with the potential for several weeds. The problem is that this is an inexact measurement. It only measures the normalized difference of vegetation indices, which doesn't tell you whether this is water hyacinth or giant salvinia or some other unknown weed. But it does tell us that there is vegetation on this, and we can apply that over an entire country like they did here with South Africa. I'm going to plug a talk for one of my cooperators coming up, and that's the Usman Mohammed, who is developing much more specific signatures for water hyacinth and our agents. So instead of necessarily using these inferred vegetation indices, we can actually develop spectral signatures that will tell us from an image whether or not we have water hyacinth as opposed to other floating vegetation, whether or not we have the weevils, and whether or not we have the plant hoppers. So this is just a schematic. Again, he is going to give us lots of information ahead of time. But very interesting stuff. So before you completely glaze over on this particular graphic, a really exciting thing just came out, which is the entire genome of water hyacinth has been published as of September of this year. And this is particularly exciting, not just because we can, have an entire genome for an organism, but we can start boring down into that genome and understanding why water hyacinth is such a menace. And one of those things is that if we look at water hyacinth, it has several adaptations within its genome that make it particularly efficient at a couple of things. One of those is light harvesting. So if we look into the chlorophyll genome, we find several duplications of genes that allow it to harvest light in an incredibly efficient way, which allows it to turn, remember, plants use light to create energy for itself. So it can put on biomass like nothing else, especially in eutrophic waters, which explains some of these data that we've been seeing since the 80s. And we just measured again in several different places that water hyacinth is incredibly efficient at taking in CO2 and making biomass from that. So probably much higher than many of the things that you would see, including like corn and other very high productivity organisms. It also helps us understand why water hyacinth interacts the way it does with its herbivores. So again, don't glaze over, stay with me for just a second. So the salicylic acid pathway and the jasmonic acid pathways are two ways that plants really interact with their herbivores. They upregulate these defensive compounds so that they can create an environment in which things don't want to eat them. So plants have all of these secondary compounds that reduce herbivory. And that's this evolutionary arms race between a plant and all of its herbivores. Again, water hyacinth had a whole genome duplication event, which means that they have about twice as much, if not more, of these compounds than other plants do. So again, really fascinating how water hyacinth has evolved to really create a lot of energy and get rid of its enemies. And again, we can possibly use this to not only understand why things work or don't, but also potentially exploit it in different formats. So really exciting and a very big point for future research in that sort of foundational understanding role. Okay, we'll get out of biochemistry. Sorry, thank you for going along with me for a second there. And let's talk about improving and providing best management practices. And this is where I'm spending a lot of my time, which is how can we incorporate all of these various methods for controlling water hyacinth in the most efficient way so that we can use our chemicals most efficiently and make the best use of these effective organisms. One thing that I would be remiss in not including in this talk is that our chemistries for water hyacinth, and especially for aquatic use, have become more selective. So we have less non-target impact, especially on water lilies and floating plants, than we have in others. Thank you. And then many avoid damaging those rooted aquatic emergent plants. So again, water lilies, cow lilies, those sorts of things. And Patrick Moran and his colleagues showed that many of these integrate well with Megamelus scutellarius. So one of the reasons we developed Megamelus scutellarius is that when you spray a plant with Megamelus on it, those insects can easily hop off of it. And it is a dramatic improvement over diquat and 2,4-D, which were the primary modes of chemical use. So the problem with all of these new chemistries, though, is that they come with new patent chemistry prices. So as opposed to 2,4-D or glyphosate or diquat, they are incredibly expensive, but they can be used at much lower concentration rates to get the same level of control. So really interesting work on the chemistry side and how we can incorporate those with our insects. And that brings about the need for these mesocosm studies looking at integrated management. So when we often work with in aquatic invasive plants, we do these studies in mesocosms, which can be tubs. In this case, these are coffin liners. And we can see no biocontrol on the top. We get a little bit of browning, and that's just due to overwintering. If we add biocontrol, we get some reduction, although not much. If we have operational and reduced operational rates of 2,4-D, we get death in the tanks and water hyacinth doesn't come back. This, of course, though, doesn't translate onto a highly changing lake environment. And that's why we installed this integrative management on the fourth largest lake entirely within the United States, which is Lake Okeechobee here in Florida. So these are permanent anchored vegetation barriers, keeping in water hyacinth and a whole bunch of other treatments. So we have reduced rate 2,4-D, same kind of setup as we do And then these are going to be monitored aerially and also with pulling out random plots for biomass and counts. They're spread all across the lake. It's an incredible amount of work that my crew did, but hopefully we should be getting information out of those soon. And that's really important because now we have real life data. Thank you. And we need real life data because this is a real life problem for the entire globe. So we are not the only ones dealing with water hyacinth. No continent is unaffected by water hyacinth except for Antarctica. And this illustrates the fact that we need an international working organization for water hyacinth that includes not only the people in this forum, but also folks from the global south who are devastatingly impacted by this weed. So I want to thank everybody, and I'll be around for questions at the Q&A session. Thank you.
Thank you, Melissa.
And I'll stop sharing.
All right, so our last speaker of this first section is Rodrigo Diaz with Louisiana State University.
Good afternoon, everyone. Can you hear me?
Yes, we can hear you, and your
Awesome.
slides look great. Thank you.
Thank you. So yes, first I would like to recognize that what you're going to hear is what comes from a conversation with several colleagues and friends that we have been dealing with the giant Salvinia. So I would like to recognize definitely their contributions, and you'll hear them throughout the presentation. So let me move the next slide. Click. Next, awesome. So what the dealing is, giant Salvinia is one of the fastest growing aquatic weeds, and still is amazing that to this day that you can see these ponds that just have Salvinia on the fringes, and it will take a couple of weeks, if not days, to see this pond just completely covered with giant Salvinia. So for me, it's one of the most fascinating components of the system, the speed of the growth of giant Salvinia in our systems. Now, what we know now is that we have the distribution of giant Salvinia. It's definitely becoming problematic also in temperate regions. I would like to mention the Caddo Lake and also in South Carolina with our colleagues in Santee Cooper. In temperate regions, this weed is becoming a problem. And as you can imagine, a vast areas of Salvinia is managed with herbicides, but there are limitations, especially when you try to get those applications in those forested wetlands. So once it gets in the system, Salvinia just becomes this recurrent problem. Now, we have the weevil. Here I would like to show the Cyrtobacus salvini, thanks to Alco, Francesco, and Julie for giving us those introductions. This is a beautiful weevil from Brazil. Adults feed on the young birds, and the larvae tunnel the rhizomes. And something important is to highlight the tropical origin and the importance of the larvae as a major source of damage. Now, in terms of populations, thanks Al for the success stories that you show with this weevil, but we also got the opportunity to release this weevil in Puerto Rico a few years ago. And you can see in this picture in the tropics, and I like the 2 years that you mentioned, Al, it was the same thing. In 2 years, we clear and reduce the densities in Las Curias. The same story happened in Cameroon, thanks to a collaboration with Rhodes University. You see these massive systems going from massive dominance to almost complete control. But the point that I want to highlight here is that, as you see, eventually the system reaches kind of these lower densities of Salvinia in the system. And it's fascinating because, for example, this is what I would like to call success. Just, I guess, was 3 weeks ago, Flaminia and Dane from my laboratory went to Puerto Rico. And what you see in Las Curias is this. Few spots of Salvinia, and that is definitely completely covered with weevils. So this is what we're calling success, right? It's not eradicated, it's just in low densities and completely covered. So a few lessons from these tropical conditions, right? And something that I invite you to think about this, especially the weevil is following the plant in these fringes, and the densities are high enough to exert damage, right? But think about also the temperatures, ideal, beautiful temperatures for activity of these weevils year around, right? But definitely, please think about what we can manipulate here in temperate and subtropical regions, thinking about spatial distribution and densities. Temperature, of course, we cannot change. Now, let me show you the story a little bit, what's happening in the United States. I want to show you this survey that we did. Here will be Louisiana, you can see here, the southwestern area. And if you see a satellite image, you will see this complex of water bodies from canals, small ponds, large ponds, all interconnected. So this huge area of 8,000 hectares, we got a chance to sample and collect density of weevils, which they have been released for almost 10 years. And these, again, natural densities that you're going to see. In this graph, I want to show you first on the left, you see the Salvinia cover in these locations and the time. And you can see the drop of Salvinia cover in June. If you go to the graph on the right, you see Salvinia weevils per kilo, and you see the 40 in June. So what we're using is, this survey, like, whenever we see 40 to 50 weevils per kilogram in May or June, we can tell the folks that, you can see these drops of giant Salvinia again in these subtropical areas of Louisiana. Now, something interesting about this survey is the patchiness of the distribution of the weevil in the landscape. You see those dots? They show the densities in June. Some areas you have 7 weevils per kilo, and just a few miles, a few kilometers away, you have densities of 80. I invite you to think about what it will mean in terms of, what area is going to crash versus what areas are going to give you trouble. So that's something important we want to highlight. And this is, the idea that this weevil is very patchy in their distribution. So that has been fascinating. And what you see in the field, of course, is the brown patches with high activity versus those green patches where have less densities of weevils. Now, so this is what probably is the new normal in South Louisiana, where you have these crashes of Salvinia in some locations, but other locations nearby, you see more other Salvinia basically under less pressure, and that system kind of continues in the landscape, kind of these patches of control and outbreaks, right? And something important I want to highlight is this: just because this is the new normal of natural fluctuations, doesn't mean that we cannot do interventions in June. I still think that there is an opportunity to improve our management if we do some early interventions in the system, and we're going to hear about that. So this is a situation in the subtropics where you have these outbreaks of Salvinia early, the weevils come and they crash, but there are other areas later in the summer that you will see. And right now, in fact, just a few weeks ago, we know that there are infestations in November of Salvinia. So this is what I mean, that this kind of localizes success of the system. And of course, it's driven by the temperatures that allow weevil activity. Now, I want to take you now to the most tougher area, that is the temperate regions in the areas of Louisiana. And in this graph, what it shows is the average number of larvae of weevils per kilogram. And then you have Cross Lake in the north of Louisiana and San Gabriel in the south. And what you notice is that in San Gabriel, around late February or early March, you see larvae in the samples, right? It's early start of the population. But if you see in Cross Lake, it's later. They take longer time for that larvae to show up. And then you already start thinking about issues with right there, disconnection of the populations in these north and southern locations. But we know that we have a team of people, including the U.S. Army Corps and several agencies, exploring if there is opportunities to look for cold-tolerant populations where we can make a difference with the management specifically about improving the survival in the winter. Now, I want to show you some success story from Judson Riser in the Santee Cooper area, which is in South Carolina, right at the edge, right? And this is you see the location. And what we're finding is that Johnson was able to release some weevils in 2023, and then he saw some establishment in these areas, which is fantastic, right? You can see the browning in August 2024. But the approach, as you can imagine, is, where we can strategize the releases of these weevils in these tough areas, right? So it seems to be that maybe we can think about putting these weevils in these hotspots in the periphery, in this kind of at the edges of the infestation, in such a way that you build numbers, at least especially where they should be. So it's very promising, this idea that maybe we can be aiming at putting the weevils in these hotspots. And of course, Salvinia is going to be the middle of that lake, for example, and this is where herbicides may play a better role. So it's very interesting to think where the weevils have a better chance to have an impact in management in these locations. Now, so if I can briefly explain the story in the temperate regions is this, right? That you have these outbreaks of salvinia, the increasing numbers, and the weevil just struggles, if you see this yellow line, to catch up, and then eventually they don't get the densities that they need throughout the landscape. So it's very interesting, again, to think about, especially and in terms of the densities, how we can strategize to make them more effective, perhaps in the periphery, right? Focusing on those backwaters or hotspots. Now, and this is the graph showing temperatures, and that's basically to highlight, Lake Charles will be our southern Louisiana locations, and the idea was to show the longer period of activity of these weevils in southern, more subtropical regions, while in more temperate regions, we do have a shorter window where the weevils can help us with management. Now, I want to mention a new issue that we have in Louisiana, which is the group from St. Tammany Parish are finding that there is huge numbers of mosquitoes associated with these mats of aquatic weeds. So, I mean, it has been fascinating. We know that Mansonia has been reported in the literature, but now they are detecting tremendous numbers of these mosquitoes. And you can see the larvae attached to the roots, and you can see the salvinia in the right-hand side with egg masses of these mosquitoes. So they are creating a new problem for the abatement district. But what is interesting is also the public pressure to address this issue. And that got us thinking with Kevin and his group to say, well, is there a way that we can help accelerate biological control? What is the topic that we're talking, right? Inundation approaches and how we can brainstorm ideas to actually make these agents work better for us. So this is the next part of the presentation is thinking about opportunities, what we can do about this. And one of the first opportunities, actually, what already Melissa mentioned, is the idea of remote sensing, right? If you have the map of these wetlands in southwestern Louisiana, we need to find salvinia, this idea of early detection. And I invite you to think the complexity of the habitats that we deal with, right? You have salvinia in these small ponds and large ponds and canals. So especially we need to understand where it is early in the season. So as I'm glad that Melissa got a chance to introduce all this topic, but thanks to Rhodes University, and we're using Google Earth Engine to develop this remote sensing capability to detect these aquatic weeds. And as we recognize that we have to use other type of sensors to detect at a finer resolution. I would love to know where are those brown patches, right? Where I can use them. So there is a tremendous opportunity. And something important is this, we need to make it operational, how the people have access to this content. And this is because, we need to use web applications. And that's something that Google Earth Engine is allowing us to do. Any person can access these applications and get the data, get those maps to know where these infestations are. So it's very promising. And I'm glad that we're kind of bringing all together these ideas of improving detection. The other idea is augmentation, right? In terms of, I need to know, once I know where my salvinia is, I need to be putting weevils in those specific hotspots, understanding especially, again, where they are and how they need to be following closely salvinia, right? And with that, you guys know we need to improve our capability to do mass rearing. I love that Julie showed all those points around the lakes. I wish to have that capability around all these lakes. But the idea is that, right? We need to improve our rearing operation. And not only that, but the ability to know how we do the releases, whether there is a most effective way to put those weevils in those locations. And what is fascinating is this, once you have those field nurseries, we need to use them. And that's the idea that in the case of the Salvinia program, we're definitely shifting our mentality to say, hey, we give you weevils, you need to go back to your wetland, find the brown spots and move them, right, to the green area. That is going to definitely accelerate the process of colonization of these weevils. But, it's very hard for people to go in the boat and move them. So we're thinking about opportunities. And I like the idea of drones, and I like to recognize Kevin's idea is this. We do have the idea that, drones can detect color, and there is these actually piercers that you can use and attach to the drones. And it's not far-fetched that we can see the right-hand side weevil with Salvinia weevils being taken to the locations they are needed. So there's plenty of opportunities, and we do have scientists. I'm thinking about Ivan Grijalva, who is a new scientist working with remote sensing capabilities to come up with ideas to improve this movement of weevils as they are, again, making the use of field nurseries, right? Now, this is a word that I would like to recognize of Alex Gafke. You know, we need to use attractants, and you guys saw the publication from the air potato beetle and the use of semiochemical lures. How about calling the weevil to locations where they are needed and say, hey, you know, we know that they're in the landscape, we pull these lures, as that's the picture in the air potato, and calling them where they are needed, right? And that, I think, is an idea that is fascinating for me, to have that tool to manipulate numbers. And as you guys know, this is well documented in agriculture and forestry. And I got this picture from the internet about this septa that you can call rice, weevils, and so on. But to make it practical, needs to be, there's a commercial product. We need to have developed these technologies, but I think may help us with management. Awesome. We're closer to the end. Is the final message. So this is it. So I'm very excited that we do have an opportunity to improve the job of the Salvinia weevil. I wish to have all the agents of the hyacinth, but we don't. For the Salvinia program, we do have one weevil, and I think we can improve their management. One of the messages that I want to share first is, you know, Salvinia is still a major problem, and stakeholders are asking us for solutions for management. The weevil definitely needs some help, and let's think about, I invite you to brainstorm this idea, especially in a water body, and also the densities that how we can change those to improve this colonization, improve this activity within a water body. And I still think that, you know, these extra interventions that we're gonna perhaps do is they're gonna have higher costs, but I still think it's gonna be cheaper than bringing harvesters or spraying herbicides with helicopters when you have 100% cover. So and with that, I want to thank you for your attention.
Thank you so much, Rodrigo. That was great. So now I will turn it over to Jen to ask some questions of our presenters.
Thanks, Melissa. I am going to turn my camera off just for bandwidth issues. So we've had quite a few great questions already in the Q&A box, and many of them have already been answered, but I think it'd be great to have some of these answered live. So we have a question from Jason for Julie, and I believe, Jason, hang on until a little later in the session and you'll hear more about parrot feather. But Julie, what species is most effective at controlling parrot feather in cooler temperatures?
In South Africa, we've only released one biocontrol agent, which is the chrysomelid leaf flea beetle called Lysathia. It's busy. It's being worked on in Argentina to be named. It does work at high altitudes and cold winters, and we do see it's a pretty resilient tough beetle. But I think, yeah, as Melissa said, that Philip Vail will be talking about it a little bit later.
Great, thanks, Julie. And one question we very often get asked in these summits is whether our biocontrol agents are safe and whether there's impacts to non-targets. And I suspect this entire panel could answer this question. So is there somebody who would like to particularly answer a question about the safety of our biocontrol? Phil, I know you put a paper into the answer section, but if somebody wants to take it away, go for it. Maybe I should call on somebody. Melissa, you unmuted. Would you like to do it?
Oh, it's because I was going to tell Al he had to answer it. Well, Al did actually show a really nice diagram of the flow, right? So we start with our international cooperators. We go over to where the weed is native, and we collect herbivores on it and bring those back over. And we're specifically looking for specialists. So biocontrol, though, can be insects, mites, and other arthropods. It can be plant pathogens. So there's a number of things that can be used. But what we're looking for are things that specialize on that plant. And a good example that I use for the lay public is that if you think about a specialist, like think about monarch butterflies, they can only live on milkweeds. That's a very North American perspective, but that's a good example that the North American audience usually gets. So that's what we're looking for. And it's pretty conservative biochemically to be a specialist. You don't have to come up with all sorts of extra things and pathways to break down those nasty plant chemicals. You can only break down the plant chemicals that are in the target, in your preferred host plant. And so then we test it. We usually do no choice tests to start, and then choice tests if we need to, which is totally unrealistic. And in most examples in the real world, you're never going to have a time when the insect doesn't have any option except to eat its non-preferred host. So we usually go through that, and then it goes through once we are, as scientists, are satisfied with the specificity and impact of a particular agent, it gets, at least in the United States, it gets shunted into a extra governmental multidisciplinary review, which is the technical advisory group. That's an offshoot of APHIS. And then it goes through a permitting process. And in all of these examples, if they are approved in the United States, they, new ones anyway, go through a public comment process even. So the public has an opportunity to comment on those. So that's the generalized safety testing. But we start with very close relatives and then move outward from there. So it can be anywhere from 30 species that we test to, I have a test plant list that has over 100 species on it. So it just depends.
Thanks, Melissa. Here's a question for Al. I hope, Al, you can unmute, okay. You showed throughout the 3 decades of biocontrol for water hyacinth, how often did you need to add the weevils and how many to gain that control?
Hopefully you can hear me. We only added the weevils early in 76. We developed some rearing colonies and established them. We never really continued to distribute them for maybe about 3 or 4 years. We were moving them around Louisiana, and then it was sort of a free running system. We had some examples very similar to what they saw with Megamelis, where the weevils were inundating some schools in the Atchafalaya Basin, close to new mercury vapor lamps that they put in. We sent Gary Buckingham down there to look at it. So we never really augmented them. We just let them establish and free run. But there's a vast amount of water hyacinth down here, so it took a long time for the population to build up.
Thanks, Al. And here's a question again for Julie. How did you manage and in parentheses, I'm assuming some negative feedback from public about the huge increase in the number of insects?
Yeah, that was quite something that we had to deal with. So we've got a really good social media platform on Facebook as well as Instagram, and then we also send a lot of information through to the local newspapers in those areas. So obviously when these Megamelis started swarming, we were getting calls saying these bugs are in our house, they're attracted to our lights at night, we can't sit outside. A really big thing for South Africans is a braai, which is a barbecue. You know, people couldn't barbecue, which it's our national sport. People would go to restaurants and get these bugs in their pizzas, and so it really was a pretty big issue, and happened rather it wasn't anticipated. We'd never seen this boom before. But what we could do through our social media campaigns and all these communication channels was assure people that it would be short-lived, because the insects, the megamelis, wouldn't be able to feed on anything else, so that they weren't going to attack their garden plants, their crops, their rose bushes, and that they would die quite soon. And along with it would come the crash in water hyacinth, so meaning that they would get access back to their dam, their lake, essentially. So yeah, and then it happened again 2023. And then people were a lot more accepting of it by saying, oh, wow, okay, these insects are exploding. This obviously means water hyacinth is about to disappear. So yeah, a lot of publicity and communication.
Wonderful. Thanks so much. I don't think we have time for the one more question that went into the Q&A. If Julie, if you wouldn't mind answering that online, that would be great. Thank you. I will hand it back to Melissa.
Great, thanks, Jen. Okay, so now on to our second section, which is focused on rooted aquatic species. And our first presenter for this section is Rob Boucher with Agriculture and Agri-Food Canada. Are you there, Rob? You are.
Sorry, I'm having bandwidth problems. Just going to leave camera off. Can you hear me okay?
You know, I can hear you, yeah, maybe a little bit of cutting out, but for me, your slide is just like the top left-hand corner. Jen, are you seeing that as well?
Yeah, we can see implementation of biological control, not the rest of the word.
Oh, but you can see the middle of it? I don't know. It's, yeah, the computer's just going to melt.
So your audio is cutting out a lot now too.
Maybe could we flip and I'll just try and reboot? Could we flip to the Jen talk?
Okay, yeah, I think that sounds great. Well, not to put you on the spot, Jen, but are you ready to jump ahead and present? And maybe we can hop back to Rob at the end.
Yes, that's no problem. Rob, if you could just stop sharing, and
yeah.
Hopefully I don't have any problems.
Okay, so we're going to switch it up a bit, and our first presenter will be Jennifer Andrews with Washington State University.
You got control? Okay, good.
Yes.
Yeah.
We are seeing your notes page, Jen.
Gosh, sorry. We thought we had it figured out again. Here we go. How is that?
It looks great.
Okay, wonderful. Thank you. Well, thank you, everyone. So far, this has just been a really great summit, and I'm excited to share our work on flowering rush. I just want to make sure I can see you if you need to call me, Melissa, and I can. Okay, so this is a project that involves a lot of different partners, and I do want to make sure I recognize that this is not something that is solely my work. In fact, a lot of this is other people's work. So I will talk about each person in a minute. Just to give you a little background on flowering rush, it's Budamus umbellatus, and this is a freshwater aquatic invasive species. It's colonizing wetlands and sort of slow-moving rivers and canals. We find it in irrigation ditches. It can also be found in fairly faster-moving water. So I always think with flowering rush, you shouldn't dare it to tell it what it can't do, because it really can grow in so many places. We have both a diploid and a triploid cytotypes, and it reproduces differently with those, and I won't go into it. We also have some ability. So here in North America, we have several different growth forms, and that really impacts how we're going to be able to do biocontrol. So it can grow from fully submerged, where the populations are completely underwater and the leaves never emerge out of the water. And in the West, we have a lot of our populations that are fully submerged. It can also grow partially submerged, which you can see in the photos here on the oops, let me do this one here. You can see over here on the left, and then it can grow fully emergent as well. We don't see this as often. This is the current flowering rush distribution in North America. So it does tend to really be in the temperate area of North America, the southern border of Canada, and the northern area of the US. And it doesn't look like there's a lot of populations, but once it enters a water body, it really can move fairly quickly. And so it's a pretty big issue. One of the concerns about flowering rush is it's incredibly difficult to control. So there's been a ton of research and a lot of people putting great effort on the ground trying to control flowering rush. But it's tough to control with herbicides, and all the other techniques are also, only moderately successful, and it really seems to be site dependent in terms of success. And so we really started thinking about, flowering rush is going to be this hard to control. What are our other control options? And back in 2012, 2013, we started thinking about whether biocontrol could be an option. So one of the cool things about this plant is that it's the only species within the Butamaceae family. And this is really increases the likelihood for us to have finding a host-specific biocontrol agent. So it looked like a great candidate. So we formed a consortium in 2013, and we've been working very closely with CABI Switzerland and CABI UK in order to find these, to find potential biocontrol agents. So our current project researchers and collaborators include Patrick, Sarah, and Phil from CABI within Switzerland and the UK. We work very closely with Natalie West, and John Gaskin has since retired with ARS. Melissa, who you've met today, Bryce is also very involved. He's in Missoula County. Canada is ahead of us in terms of getting insects on the ground, I'll talk about. And so we're working with Rob Boucher and Dave Ensing, Nathan Harms from the Army Corps of Engineers, and then Janine Brooke from British Columbia. So all of these people have been actively working on this project. As I mentioned, we have six, so we have different genotypes, and there are 6 known genotypes currently in North America, but we're really dominated by 2 different genotypes. In the West, you see all the blue marks. This is genotype 1, which is a triploid. And on the East Coast, in Midwest area, there's more genotype 4, which is a diploid. So they're reproducing differently. And this plays a role as part of our choosing biocontrol agents. So there are currently 4, I actually realized I shouldn't say 4 potential biocontrol agents. There are 3 potential biocontrol agents and 1 approved biocontrol agent that I'm going to speak about. And I apologize, I do not know where this photo disappeared to, but this is a fly. So hopefully it will show up on my next screen. Oh, here it is. Okay. So this is a brand new fly that we're just beginning to do some work on. So the larvae are mining the leaves, and that includes fully submerged plants. And there are 2 generations a year. There isn't that much known about it yet, but as I said, this is just getting off the ground. And the reason we're really excited about it is it may be very important for managing submerged plants. And this is something that we're really focused on now in terms of the research. The other one we're excited about for potentially for fully submerged plants is a white smut. So they have the potential for high damage. They do quite well in aquatic habitats. And this is a picture of what a flathead lake. So these are all plants that are fully submerged. And so although our arthropod biocontrol agents that we have been looking at may not do so well in here, the pathogen may have the ability to really knock back these populations. So it's been a bit challenging to find a strain of this white smut that will attack our 2 primary genotypes. And so this is an extremely smut. And so we've really struggled with finding a strain that can be damaging to our genotype 1 and 4. But thankfully, back in 2021, they finally found a strain in Romania. And so right now where we've been having some challenges is with reinfection, and more work is needed with this before host specificity testing can begin. So this is one that still has a couple of years work to do. The third potential biocontrol agent is the Phytolyromyza ornata. So this is another fly. The larvae mine the leaves and the flowering stems, and again, 2 generations per year. So initially, when we started looking at potential biocontrol agents, we really weren't sure that this was going to be very successful because it wasn't really getting down into the rhizomes of flowering rush. But Patrick continued to do more work with it and was finding that after exposing plants to a single female, that plants were wilting within 2 to 3 weeks. And so this really escalated its interest to us. And he has since conducted some biomass work showing that within 2.5 months, you can see a 37% biomass reduction. And when you increase that to 4 months, it can be up to 68% biomass reduction. And I know Patrick probably has some newer data on these sorts of things that he has will hear about soon in the future as he analyzes that data. So in addition to this, he's been conducting no-choice development test. So this is an extremely host-specific fly. It's 40 different test plant species have been exposed to the fly, and there has been 0 development. So the fly only wants to attack flowering rush. There is just a few more test species that need to be added in 2025, but we are planning to develop a petition for the fly this coming spring, winter and spring, and hopefully we will be submitting a petition in 2025 for this fly. So the last one I want to talk about is the one we're furthest along on, and we're pretty excited about where we're moving forward with this one. So this is a weevil, a aquatic weevil, Bagus nodulosus. Both the adults and the larvae will affect flowering rush. So the larvae feed in the leaves, and they will feed on the top part of the rhizome. And then the adults are also creating this damage, which you can see in the top right-hand picture. So the larva will leave the leaves and swim to other plants, and that's an interesting characteristic and influence some of our host specificity testing. And what's great is that the adults can survive for multiple years, and so they are able to lay eggs over several years, which really increases our chance of populations building up. So again, host specificity testing for Bagus nodulosus demonstrated that it's extremely host specific. Patrick conducted no choice oviposition and adult feeding tests for 4 years between 2016 and 2020, looking at 40 different test plant species. And there was no oviposition on non-target species. So again, females are just not interested in laying eggs on flowering rush. There was a little bit of feeding on 3 species, but it was really considered significantly less than flowering rush, more just nibbling. Now, because the larva will leave the plants, there is an opportunity for the larva to choose a host. And so this testing whether the larva will, when leaving a plant, be interested in other non-target species was conducted between 2018 and 22. So how Patrick did this was, you can kind of see an image here, exposing 3 larvae to the cut leaves of a test plant or the control plant for 5 days. And then you determine whether did the larvae survive or did they die. So 43 test plant species. Wow, okay, I better hustle. Thanks, Melissa. 43 test plant species were tested, and only a few survived on 3 non-North American species, but they all died in further testing. So we feel really positive about that, the beetle being safe. So in terms of impact experiments, the weevil looks to have a great impacts. It can reduce biomass. And I know these are some photos from this year that Patrick sent me. They are probably still analyzing the data, but you can see in untreated pools how much more robust the plants are compared to the treated pools. So we put the petition in for the weevil in 2022 in April. Canada approved it within several months, and we just got approval for the United States in October 2024. So we're really excited to move into the next phase where we will take the weevils out of quarantine. So our next steps for this is rearing has been done in quarantine in Sydney, Montana by Natalie West and her team. And here in Montana and Washington, we are establishing rearing ponds and pools with an intention to start trying to rear weevils next summer in order to have larger numbers for release in the future. And CABI will continue to work on impacts and rear insects for us as well. So in Canada, they released their very first insects in this year in Ontario, and they're continuing doing outdoor rearing tanks and also quarantine rearing as well. So we're really working in collaboration to figure out best rearing techniques. Now here in the US, we've been establishing pre-release monitoring systems. So our initial releases will occur in Flathead Lake in Montana. We're surveying sites in 2025 to find good places for us to do the same work in Washington. And then we have been conducting this data collection, and this has been in conjunction with Melissa Bryce and Natalie, and you can see what that looks like in these photos of how we're trying to understand what populations look like before release and then after. And I just wanted to thank, we've had our generous funders. We've had a great and really supportive group of people who have provided funds for this project. We are always looking for additional funds, especially with some of these newer potential biocontrol agents. And so please contact me if you feel that you might be able to contribute or would like to be involved in some way. And with that, thank you.
Thank you, Jen. Perfect timing too. Okay, so I do see Rob is back on. Rob, would you like us to go to Paul next, or would you like to try again?
We could try again, I guess. It's,
Oh, your audio sounds much better.
oh, good.
Oh, maybe I spoke too soon. I think I spoke too soon. Your audio is still breaking up pretty badly. So I think we should maybe go to Paul.
Okay, can you hear me now?
Oh, yeah, now I can hear you. Okay, would you like to try and share?
Sorry, go ahead with Paul and I'll try another reboot.
Okay, sounds good. All right, so we are going to jump ahead again, and our next presenter is Paul Pratt with USDA ARS Invasive Species and Pollinator Health Research Unit. And your slides look great, Paul. Take it away.
Thank you. Can you hear me okay?
Yes, we can hear you great.
Yeah, my name is Paul Pratt, and yeah, I'm the research leader for the Invasive Species and Pollinator Health Research Unit. Figure out how to advance the slide here. There we go. So the mission of my research unit is to develop and transfer integrated biologically based approaches for the management of invasive species and the improvement of pollinator health. And our team is actually spread across 2 locations. We have 5 scientists in Albany and 4 scientists in Davis. So our research team consists of 2 scientists that work on honeybees. That's Dr. Fine in a vacant position right now. And then we have 2 plant ecologists that focus on aquatic plant biology and management. And that's Brenda Growell and Jens Beets. And then we have 1 scientist that develops insect biological control. It's Brian Hogue. And then we have 5 scientists, including myself, that conduct weed biocontrol research. So Patrick Moran, Brian Rector, maybe a newer face to some of those on the call or the meeting today is Dr. Rebecca Mueller, who's a microbiologist who joined our team a few years ago. And then I'll also note that Link Smith, although retired, continues to be a very productive member of our research team. And before I get too far, I would be remiss if I didn't list many of our important collaborators that provide intellectual and financial support of this research. And I'll specifically identify FUEDE, which is the Fundación para el Estudio de Especies Invasivas. I think we have a few folks on the call today from FUEDE down in Argentina, and then INE from Uruguay. And then I'll also share the U.S. Army Corps of Engineers was instrumental in funding some of this research and has been a collaborator from the beginning, Nathan Harms, Alco Francesco, Ian Knight, and others. So moving on then, and kind of giving a little bit of background about the environment and the situation, but water availability, like many places in the Western US, is a strong driver of political, agricultural, ecological decision-making. And nowhere is this more apparent than in California's Delta, where two of the state's primary rivers, the Sacramento and the San Joaquin, converge in a network of wetlands, canals, sloughs, and ultimately outflow to the San Francisco Bay. So this confluence represents the largest freshwater estuary on the West Coast. It provides irrigation water for 1.2 million hectares of cropland. It's the source of drinking water for 25 million people, many of those living in Southern California. And at least from my last check, is the home of around 56 rare, threatened, and endangered species. So like many places around the world, and we've heard a lot of examples this morning, the combination of abundant water, nutrient runoff, commerce, recreational boating, and the dumping of a few aquaria from time to time has resulted in this proliferation of aquatic weeds in California's Delta. These weeds disrupt the conveyance of water to agriculture for human consumption and further endanger at-risk species. A few members of our research team have worked diligently in developing tools for improved management of aquatic weeds, and that includes the development of biological control. And Dr. Patrick Moran was highlighted earlier in previous talks. So two of the more problematic weeds that we work with here in California, and specifically in the Delta, include Ludwigia species and alligator weed, which has also been highlighted previously. So the exotic Ludwigia species that I'm clumping in together for the purposes of this talk include Ludwigia hexapetala, Ludwigia grandiflora, Ludwigia peploides, subspecies montevidensis, and then also subspecies peploides. So this species complex generally invades wetlands in the Pacific coastal states of Washington, Oregon, and California, as well as the southeastern and southern states. Alligator weed is in, totally different family, Amaranthaceae, and also invades wetlands in the southeastern United States, is found north along the eastern coastal states all the way to Maryland, but on the West Coast, it only occurs in California. I should say it only occurs in a weedy fashion in California. So although distantly related phylogenetically, both of these weeds share a lot of similarities and have similar qualities. So they're both native to South America. They both have native North American congeners. They're perennial forbs. They're aquatic, but also oftentimes the semi-terrestrial lifestyle, in that they are oftentimes rooted on the edge of the banks, but can then definitely spread across open water on the surface. The reproduction is primarily asexual, as they both root at the nodes and fragment easily. Their leaves are narrow and elliptic. Stems are prostrate and form mats as they grow across the water, as I mentioned, but they're both difficult to control chemically, alligator weed more so, as Al mentioned in his talk earlier. And they both generally invade the same kind of habitats and compete for similar niches in the Delta. So it's not uncommon for our stakeholders, as kind of is indicated by these photos here, that they're easy to confuse for the untrained eye. And as mentioned above, both the Ludwigia species and alligator weed have U.S. native congeners on a critical research hurdle is to determine if natural enemies possess this section level host specificity. And, we need to ensure that they're not going to attack native species that are found within the same genus. So another additional hurdle, of course, as has already been talked about by Julie previously, is determining if natural enemies are adapted to California's climate. We've learned from sad experience that sourcing agents from one region of an herbivore's native range, it rarely matches the entire geographic range of a widely distributed weed. So turning to the Ludwigia project specifically, addressing the first hurdle of identifying host-specific natural enemies involved surveying for natural enemies in South America, and specifically Argentina, Uruguay, and into Brazil. So this was done in collaboration and was led by our colleagues at FUEDE and supported by folks at INYA in Uruguay. And by 2019, a large list of candidate agents was developed, but many of these were known generalists and fed across several plant genera and even families in some cases. So a much shorter list of leaf feeders was prioritized. Was prioritized, and we started host range testing. But also we were quite interested, and you might see them over here on the right-hand side. There's a group of Tyloderma stem miners that were also really interesting. So ultimately, we brought in 7 insect species and ported those for host specificity testing here in Albany, California. The first 4 species tests included those for a thrips, Lilithrips ludwigia, a flea beetle, this small little weevil here, Pseudaloetes bosque, and then also semi-aquatic caterpillar and moth Pericles azoli. Unfortunately, the host range of these species included native plants, and in some cases, these natives supported better development as compared to the target weed. So it appears that none of these 4 species are suitable natural enemies, and they won't pass that host specificity hurdle. We were able to colonize 2 of the 3 Tyloderma species, the stem miners. We were able to rear them in quarantine, but development times and damage levels indicated that their value in biocontrol may be limited, so no host range testing has been conducted on them so far. So in the absence of classical biocontrol options, inundative releases of new associations may be a viable alternative. And what I mean by that is there is a flea beetle, Altica litigata, which attacks exotic Ludwigia here in California and at times reaches damaging population levels late in the fall. So mass rearing and inundative releases of this beetle may be an option, but it has yet been untested. The weevil or the flea beetles appear to be easily reared, but scaling up to a mass production level hasn't been attempted. And then, of course, as some others might be concerned or express thoughts about the fact that the spillover onto unintended targets also needs to be addressed and evaluated. This flea beetle is known as... So my phone just went off and said, hey, tsunami warning. All right, I'm going to keep going, but if I have to vacate the building, I'll let you know. Anyway, this flea beetle is known as the crepe myrtle flea beetle, so there could be some off-target damage. Transitioning to alligator weed then, it was first recorded in the U.S. and Alabama in 1897, but is now widely distributed throughout the southeastern U.S. The plant's been observed in, good grief. It looks like it's not in my area. So thanks everybody who's expressing concern online, but I think I'm okay. I'm going to keep going. But yeah,
you only have 3 more minutes anyways, so.
All right, I'm going to jump over a few slides, but luckily Al addressed many of these issues. The flea beetle on alligator weed is wildly successful and is a textbook example of biocontrol. But simply redistributing the weevil from the south, specifically below the line, the isotherm line of 14 degrees Celsius, hasn't been successful. And we've tried to redistribute those insects to California, and unfortunately, those efforts have generally failed. 2019, we made a release of Louisiana beetles, field collected and released in California, but they only went through 2 generations, and then everyone died. We were unable to collect them after that. There are other options of trying to find insects that'll pass this climate hurdle, and one of them is directed selection for increased tolerance to cold temperatures. Another is looking at hybridization, but my research team, and in collaboration with several others, we've considered new biotypes for the insect's native range, from the insect's native range. So going back to Argentina and looking at biotypes that might be better suited for biocontrol in California. And so with colleagues and this paper with Ian Knight as the lead author, we were able to compare development times and chill comas and critical thermal limits for 4 populations: Uruguay, Argentina, and then populations or biotypes from Florida and Louisiana. And we determined that those from collected near Buenos Aires and Argentina just did overall much better in every metric, just about every metric, as compared to the other populations, specifically those that are currently here in the United States. So it looks like we might have a winner, a population that's more cold tolerant. The other hurdle, of course, is host specificity for this insect. And there have been 95 species that have been tested for host specificity for this insect. Got 1 minute, my goodness. Long story short, the insect is wildly host specific, and we tested additional species that were never tested in the original testing effort, including 2 congeners that are native to the United States. But in addition, we tested some species that have been tested before, same relationship. So the insect shares the same narrow host range as the current biotype that's currently in the US. So in conclusion, 2 similar invasive species are the focus of my talk, but they have 2 very different outcomes in biological control research. The exotic Ludwigia species, we have no known host-specific species, and we're going to require looking at alternative tools of chemical, mechanical, and maybe augmentative or inundative biocontrol releases to help manage this weed. And in contrast, we do have known host-specific insects that attack alligator weed elsewhere, but those in the US are currently poorly adapted to California's environment. So we were able to find better adapted species, and we have an APHIS-approved insect that is currently going through and being the biological assessment is in the process of being developed as we speak. And I think Bob Pfannenstiel might be on the call. He could probably even provide a more updated status report on that insect. Our hope, of course, is that we will have this insect available for release in 2025 with the expectation that it'll improve water availability in California's Delta, as well as other areas further north of its current distribution where it performs poorly. With that, I will end.
Thank you so much, Paul. That was great. How are you feeling, Rob?
Give it a go. I'm just trying to share the screen.
I saw you unmuted. Oh, sharing. Okay, your slide looks great.
Okay, can you hear me?
Say something else.
Hello, can you
Oh,
hear
I
me?
can hear you. Yeah.
All right.
Okay, great. So we'll try this again.
Let's
Our
go.
next presenter is Rob Boucher with Agriculture and Agri-Food Canada. Take it away.
Okay, thanks very much for hanging in. So I'm going to talk about implementation of biocontrol of invasive Phragmites in Canada. Unlike some of the talks in the first section, this is one of the ones that's sort of in an early stage of development. I'd like to acknowledge my co-authors at the University of Toronto, Sandy Smith, and Michael McTavish, Ian Jones, and Jen Basie, and Patrick Hafliger at CABI. Okay, so I'm going to give you a quick outline of the Canadian program, the stages that we go through in biocontrol, and then I'm going to talk specifically about Phragmites, introducing the biocontrol agent, and then tell you a little bit about where we are at stage 5, which is really rearing, release, and monitoring. And those stages will become clearer in a minute. So the stages that we move through, there's a 7-step process, and we're really doing research to try and accelerate agents through those 7 stages. We start off asking, and this is pretty similar to Al's pipeline, just a little bit different wording, understanding of the species interactions in Canada. Is a biocontrol program justified? What's the weed doing? What sort of impact is it having? If that's decided, and this is a process that goes on through the whole research program, if it's decided we need an agent, then we move overseas exploration in the country of origin. Once we find a suitable agent that we think is host specific, it goes through biology and host range studies. And then those biology and host range studies, as others have mentioned, is to make sure that we have a safe, specific agent that only affects the target weed. It can take a long time to go through that long list of representative species. Once we get through that, we petition to regulators for the agent's release. And that's a petition that summarizes all the information that we know about the agent and about the weed and does a risk-benefit analysis. If it's permitted for release, then we move to the stage 5, which is figuring out how to rear it, how to get it out there, and what's the best way to release it. So this is a really ugly table, but I just want you to look at these on this area here is the 7 steps that I just mentioned. We're going up to step 5 or stage 5. Over here is the activities that we undertake at each of those stages, which I also just mentioned. But what I want you to notice is the number of weeds. They're at all different stages and they're moving through that process. We can have the same weed at different stages with different agents. So garlic mustard, we just petitioned for a seed head weevil. There's already been relatively recently approved stem root weevil that we're working with in the field. Similarly, colleague Rose DeClercq Float has been working on yellow toadflax. There's a relatively new agent, Rhinusa pilosa, which is a stem galler, and she's been working on another stem boring weevil since the 90s. So the same target weed can have agents at multiple stages. These are the ones that have been approved relatively recently in Canada. And as I said, I'm going to focus on common reed or Phragmites today. So just to go through that timeline, the biocontrol program was proposed for Phrag in 1998 with initial exploration of agents. 2005, the agents had been selected that were the most promising. Host range testing began on those 2 moths, Arcanera nerica and Lanicia geminipuncta in 2007. Took 11 years to do the host range testing. This was all work done by CABI in Europe. 2018, we submitted the petition for release in Canada and the US. The petition was approved in Canada in 2019, and that's where we are now with the Canadian release studies. So just to introduce the agents, there are 2 moths, Arcanera nerica, Lanicia geminipuncta. The larvae are the ones that do the damage. They mine the shoots and cause the stem wilt and mortality in the Phrag, reducing its competitive growth and reproductive output. They were pests in Europe and they can reach relatively high densities and cause this damage, so pretty good potential as biocontrol agents. Their life cycle, they overwinter as eggs. Larvae hatch out in the spring up until June and feed till the end of June, and then pupate for a couple weeks, come out as moths, fly for 2 weeks into August, and the females lay about 150 eggs under the leaf sheath, and then those eggs are overwintering again. So we only get 1 generation a year. So we got permission to release the insect both insects in 2019, and the program since then has really been focused on developing core methods, how to rear them, how to release them, and how to monitor them to contribute to basically an operational protocol for Phrag biocontrol. How do we integrate Phrag biocontrol into the other Phrag management options? So in terms of rearing, we've developed methods. This is building on work that Patrick did in Switzerland, taking stored or field harvested eggs. We can hatch those eggs out and put the larvae directly into stems and rear them through on different stems. They go through each instar needs a separate stem. Or we can rear nerica on artificial diet all the way through to pupae, produce pupae, and then we get moths and start the cycle again. But we still only get that 1 generation a year, but it allows us to do a fair push of production during the field season. We've also tested different release methods. We've released the eggs, we've released larvae in stems, we've released pupae, the pupae hatch and the moths come out of these containers, or we've actually released moths straight into cages. All of these methods work. There's just advantages and disadvantages to each of them, and the ones that we've really focused in on are releasing the eggs and larvae. So with the egg releases, the first releases we did were in 2020 of eggs glued to cards, and that was the COVID year. The eggs have been in and out of the cold multiple times in order to get permission to even get out to do field work, and none of them hatch. But what it did show us is that there was very high rates of predation on those eggs if they were out in the field for any length of time. So we developed this cup to enclose them in. The eggs are in this little container here. They hatch out, drop out the bottom, and then they move around to find a suitable stem to infest. They're quite picky at this time, and this is one of the limitations of releasing eggs. You have to get them in a spot where there's suitable stems available to them. We've also had good success releasing the larvae. In this case, the eggs hatch into larvae. We then manually force the implant the larvae into these frag stems. Frag stems then are put into these floral foams, transported to the field in multiple ways, and then placed directly out into the frag site where they will finish their instar that they're in, bore out, and move on to another stem. Quite labor intensive. Both of these methods work. So the inoculating the larvae is labor intensive, but highly effective. We get about an 8-fold difference in the initial feeding that we get around a release point between larvae and eggs. But the eggs are good. Releasing eggs is also a good method because you can get a lot of material out relatively quick, and you don't have to do all that work rearing the larvae. So we use both of these methods and are refining. As I mentioned earlier, the trick with the egg releases, you can do them in the fall, you can do them in the spring, but you really need to get them where there's those nice, perfect size stems for the caterpillars. So this is the situation since 2019. We have about 30,000 insects now released across 50 sites in southern Ontario. You can see a steady increase in the numbers as we've gotten better at rearing and better at producing them, and an increase in the number of sites. Once they're out there, of course, you want to monitor them and see what they're doing. So this is a case of an egg release canister. We monitor with a 65-centimeter hula hoop around there, assessing for larval damage in the year of release. So we want to see, are we getting these boreholes? You can get stem wilt or branching in the stems. So there's an assessment, first of all, to say, are the insects out there in the middle of the summer? And then in the fall, we do a follow-up assessment to look at the impact on the plant. And so this would be the hula hoop again. What we're finding right away in the year, in the release pod, is that we're getting reductions in the stem density and the number of panicles formed. So the gray bars here are the release plots, the open bars are the control plots, and this is just the mean on the vertical. So there's a significant reduction, which is really encouraging in the number of panicles and the number of stems that you're finding in the plot. So right away, within those very small release plots, and that's a long way to go to going to a whole frag stem, but it's looking quite promising. Once you move into the second year of monitoring, the moths will hatch out and fly around in the patch. So you've got to spread out into that bigger area and monitor it. So we go to a 4-meter long section that we go along and record presence, absence of damaged stems, and get an estimate of the number of damaged stems per meter squared. So that's the subsequent year's monitoring. When we do that, and in this table, what I have is just a list of the sites, the first 30 here, the different release methods that were used. And then we have a damage estimate in the first year, initial year, year 1, year 2, year 3. You don't need to look at the numbers so much. If the box gets darker, that's a higher number. The key point here is that we have agent persistence at over 90% of the sites. So it's really encouraging. We put them out there, 90% of the sites have insects. And we're getting reproduction and dispersal within the sites. The insects are moving around and finding new plants to infest. This is what some of that dispersal looks like in the patch. So this is the initial release point with the monitoring hoop. And in this case, you're getting larvae moving away from that hoop where they were put out. And, so it's about 2 to 3 meters away that you can see that right away. The next steps that we're going to is redistribution of agents from nurse sites. There's others that talked earlier about building up those nurse populations for redistribution. That's the model that we're taking with the frag insects, large releases to try and get recollection populations established that we can move from. To do that, we're now to the point where we can harvest stems from within a site that has a lot of insects, get an estimate from those stems, how many eggs are on them, and then we move them, just the entire stem, plant them out in these cutting devices, and this can be done in the spring, it can be done in the fall. It works best in the spring, but it does work in the fall as well. We know that experimentally because at one of the universities near Toronto, they'd actually planted frag in these nice containers here for some reason. So we were able to use those as experimental plots and do a transfer experiment right into them where we moved the stems in at different times. And encouragingly, this is what you get. The damage follows up. So the insects make it through if you move them that way, just cutting the stems, hatching out, and finding new ones. The other things that we're working on, we're obviously there's two species involved, so we're looking at the interaction between those two species. We've working with Canadian Forest Service on a pheromone to monitor the spread of the moths, looking at where they like to oviposit within the site. They're not going to work everywhere, but they will have preferences. And finally, we're doing some drone monitoring to try and can we capture the damage? So just to summarize, we have 30,000 insects released across 50 sites in Ontario. We've developed effective release protocols for the eggs and the larvae, had successful reproduction and overwintering at 90% of the sites, and we're now working on nurse site establishment for redistribution, and with a long-term goal of integrating biocontrol as a tool in integrated frag management. And I'd just like to finish by acknowledging all the people. It takes a village to raise a child. It takes an army to do a biocontrol program. So these are all people that have helped. And with that, I'll finish. Thank you.
Thank you so much, Rob. I'm so glad you kept attempting to make this presentation work because it was well worth it. So thank you.
Great.
So I will now turn it over to Jen for our second question and answer period.
Great, thank you. And thank you, Rob and Paul. Fascinating stuff. So our first question is from Mark, and it's for me. And the question is, did the release of biocontrols in Canada continue on into the US? So this is the Vagus nodulosus release. Know that the release in Canada was just happened this year. It's only one site, and I didn't get to talk a little bit about how difficult it is to rear this weevil, and so we don't have a lot of them available, and it's part of why we are doing a bunch of work in trying to put in pools and ponds in order for us to have larger numbers. So at this point, I think Canada, for their future, and Rob, you can correct me if you're wrong, will continue to be working in the Ontario area and potentially British Columbia. And then we in the US now this year that it is approved, in future couple of years from now, we'll be doing releases probably primarily in the West to begin, and then having them more available perhaps for the East Coast in several years. So, so yes, we are not sort of doing cross-border releases at this point. So Alicia, this is a question for Rob. When looking at potential hosts, did you look at frag native and non-native hybrids?
Well, yes, but they were considered, the part of the host range testing included native frag in Europe, and there's an extreme preference for the introduced frag. The moths will only lay a small proportion of the eggs on the native frag, and if they lay the eggs on the native frag, the big difference between native and introduced is the leaf sheaths fall off in the winter, and there's very high mortality of those eggs on the native frag. So it was considered. I mean, basically, it's a choice in terms of the risk to the native frag and other species of the introduced Phragmites and the impacts that it's having on those other species that led to the decision.
Thanks, Rob. And I guess I'm going to ask a follow-up question to that is, are you, in the areas where you are doing releases, is there native Phragmites there, and is that something you're monitoring for?
Yes, we're monitoring the native frag patches. They're not always available in terms of the invasive has taken over and wiped it out, but we have got a series of paired release sites where we're releasing on the invasive frag, and then in proximity there's a native frag patch, and to date there has been no evidence of attack on the native Phragmites.
That's great. Thanks, Rob.
And I mean, we're also conducting planning to conduct some experiments, additional work in support of the work in the United States to try and characterize the potential non-attack on native species.
Great. I will cover this since I'm actually a little surprised nobody's asked the question about what is going on with the FRAG petition in the United States. Both of these moths have been petitioned to a USDA APHIS. It was approved, no, sorry, it was recommended by the technical advisory, but currently it looks as though these moths will not be released in the US. Bob, I know you can't speak, Bob Fannin still, but if you want to write something more detailed into the chat, that's fine. The challenge that we have here in the US is that Phragmites, even though it may be non-native Phragmites, is holding together wetlands down in the southeastern US. And so there's actually APHIS funded projects to try to maintain Phragmites populations down in the southeast US because they are already being attacked by the Rosa, I'm not sure exactly how I say that, the Rosa cane scale, which has been quite devastating to Phragmites down there. And so it's sort of a conflict of interest where, you know, in many areas, we really want Phragmites to be controlled. But in the southeast, they're really trying to maintain populations to not lose those wetlands. So there's also some concern about host specificity with the native Phragmites here in the US as well. So again, Bob, I hope that explains what you were thinking for and how you wanted that question answered for the US. We do not have any other questions, and unfortunately, Paul did step away. It looks like he'll be okay, but it's not. It's concerning. I didn't realize there must have been an earthquake. But one question that both Melissa and I have for Paul, and if any of our panelists know where you can buy those big boat shoes that he showed in one of his images, that I think that those of us doing aquatic work would love to know more about that. So if you know where those, if you know where we can find those, we'd love to hear about it. With that, and are there any questions from the panelists? Because we are going to head into a break in about 4 minutes, but we can go in early if we have no other questions. Okay. Oh, hey, Melissa.
Hi.
Take it away.
Yeah, I think that's great. We'll just have a little extra time in our break here. So yes, we will take what will be a lunch break for some of us and maybe not for others. And we will start again at 1:35 Central Time. All right, and if anybody needs anything, probably the chat would be the best place to ask questions during the break. Great, thanks everybody. Hi, everyone. It is 35 after whatever hour it is you are in. And so it is time to start again. So our next section is on post-release monitoring. And our first presenter for this section is Christiana Aude with the NASA Jet Propulsion Laboratory. So
Can
please
you hear me?
Yes, we can hear you. And your slide looks great.
Let me just move this. Okay. Thank you for having me here today. So I'm going to be talking about mapping floating aquatic vegetation using remote sensing, specifically in the Sacramento-San Joaquin Delta. I do want to highlight that even though my current affiliation is with NASA JPL, all of the research that I'm going to be talking about today was conducted during my PhD at University of California, Merced, under a project funded by the Delta Stewardship Council. So remote sensing enables us to support field measurements with some synoptic views, and that's very important for monitoring and understanding restoration practices. I do want to highlight that remote sensing is not there to replace field measurements, and that the two ultimately provide information to each other and enable us to understand ecosystems better. For remote sensing, there are three types of platforms that we often refer to, which are satellites, airborne mounted imagers on planes, and then unmanned aerial mounted aircraft. So that's drones, UAVs. And all three of them have different trade-offs. So satellites tend to have a quick revisit time and the best geographic coverage, but they do have larger pixel sizes. And by larger, I mean they're coarser, so you can't get as fine of details. While air-mounted or airplane-mounted data sets do have these smaller pixel size, but they have longer revisit times because you do tend to have to pay for these flights. And then drones have the smallest pixel size, and once again, kind of have this long revisit time because it's an on-demand flight. So luckily for me, Paul already introduced the Delta. So I'm not going to go over this slide too much, but I am going to be telling an invasion story from the remote sensing perspective in that setting. And the two main genus level types I'm going to talk about are water primrose or Ludwigia and water hyacinth. These are two of the main floating invaders. I understand that water primrose is not free floating like water hyacinth, but because of its ability to really conquer those open waterways, we're going to consider it as a floating species here. So in 2018, invasive species covered about 40% of delta waterways, and other key invaders include like submerged aquatic vegetation like Aegeria densa, et cetera. Water primrose specifically increased coverage by fourfold between 2004 and 2016. And in fact, it's actually starting to encroach on native emergent marshes. So it's really just like, you can see in this picture here, it's actually like growing on top of the tule in this environment. And one of the ways that we know about this, besides field measurements, is from these annual high resolution airborne imaging snapshots. So this is with the hyperspectral imager, and it's on a plane, and people go out into the field and collect concurrent GPS points. And then using the actual imagery, we can map the water primrose, water hyacinth, submerged aquatic vegetation, and water, and kind of get an understanding of how these invasive floating species are increasing in abundance and where they are. So the image that I'm showing here are maps from the airborne imagery. And as you can see, by October 2016, all of these yellow patches, which are water primrose, have started to encroach on what is gray in this image, which is the emergent marshes. So really expanding, really kind of nasty stuff going on. But these flights are costly, and the annual acquisitions do miss key stages in the vegetation growth cycle. So what a lot of my PhD research focused on was understanding how Sentinel-2 satellite data can be used to fill in these temporal gaps so that we can actually capture all of these different parts of the phenological cycle. But because this hadn't really been studied much before, first wanted to see if we can distinguish between water hyacinth and water primrose at the genus level. So on the right here is an example of an airborne hyperspectral image. I'm sorry, left as an airborne hyperspectral image, and on the right is the Sentinel-2 data set. So you can see this kind of like water primrose patch in the brighter green, and then the darker green is water hyacinth. And then below are the spectral signatures of the two instruments, just an example of it to show how much more data you actually get with the airborne hyperspectral versus Sentinel-2. So that's why there was even a question about whether or not this was possible, just because you lose a lot of the information at this multispectral satellite resolution with Sentinel-2. So my target classes for classifier that I built were water primrose, water hyacinth, emergent vegetation at the community level, riparian vegetation, submerged aquatic, and NPV. And if you look at the spectral signatures from Sentinel-2 here, you can see that there are differences between all of these classes, and hopefully that'll show that that the classifier would be successful. So I used Sentinel-2 data that was but like a closest overpass to 3 of the airborne flights that we had, and adjusted GPS points collected for the airborne imaging spectroscopy flights and adjust to Sentinel-2 pixels, and then built my own random forest classifier for each one of those dates. And all 3 of the models that I built had above 87% overall accuracy for the maps. We're just going to look at some, like a comparison here. So Sentinel-2 is on the left hand at the 10-meter resolution, and then the imaging spectroscopy data at the 1.7-meter resolution. But you can see, if you look at the patches of water hyacinth, which are in purple, and the water primrose in yellow, they do match up spatially in where they are. And there are some differences just in terms of accuracy of these 2 invasive plants, because water hyacinth overall has less large patches than water primrose, but there's more in the paper about that. And then in the paper as well, I do touch on the fact that SAV can be mapped, but there are big differences in coverage due to tides. So basically this one image here in the left upper right corner was at a flood tide, and that has you're not able to capture all the submerged aquatic vegetation because of that. And this is all in this paper, so I recommend you checking it out if you're interested to hear more. But I did say earlier on, we wanted to exploit the high temporal resolution frequency of Sentinel-2 to understand more about invasion processes. And so invasive species tend to have phenological advantages over native or competing invasive species, and that can be explained by different hypotheses. So one of which is like vacant niche, so that exotic species blooms when native species are not in bloom. B is the exotic species blooms earlier than native species. C, they have a longer growing season. And D, they're more adaptable to different climate scenarios than any of the other species that they're competing with. So I want to look at what the differences were in terms of landscape phenology metrics using remote sensing between water primrose, water hyacinth, and emergent vegetation as identified in the previous Sentinel-2 maps that I showed. So to do this, I used a vegetation index. I believe someone else mentioned that in their presentation, but it essentially utilizes the near infrared and red portions of the spectrum to get at an index of greenness. So high EVI is something that's very green, and low EVI is something that's kind of brown and starting to senesce. So to do phenology metrics, I use TimeSat, which is a commonly used program in more terrestrial spaces. So I calculated an EVI for each date between 2018 and 2020, where we had Sentinel-2 imagery. Then I fit a Gaussian asymmetric fitting function to that. And then from that, I can extract phenology metrics, like where these two dots just showed up, which would be like the start of the growing season and the end of the growing season. That's defined by the left edge at 50% of the amplitude here and the right edge at 50% of the amplitude. And then you can also look at things like the length and then the rate of increase and decrease using this program. So when you look at the 3 different types, and this is just one pixel example, you can see that there are differences in phenology between all 3. And when I started to look at some study areas, and I'm only going to show results from start of season and length of season here. These are 3 different sites within the delta with different characteristics of freshwater versus brackish, etc. But water permit, which is in yellow, water hyacinth in purple, and emergent vegetation in green, and then day of year here. So the earlier the day of year is, that's the earlier the start of growing season. And regardless of year and location, water primrose was growing earlier than either of the other 2 vegetation types. And then when we look at length of season, water primrose is also growing longer than each of the other 2 vegetation types. Based on that initial figure I showed and saying that water primrose has niche breadth and priority effects in this system. But remote sensing really does give us a chance to look at these maps of starting growing season and length of growing season, which show additional interesting patterns. I'm just going to take you quickly through another thing that I saw. So there can be considered like 2 types of water primrose and emergent vegetation types. One is water primrose that grows over open water, and one that terrestrial one that is starting to grow inland. And then I'm also talking about, we have emergent vegetation in terms of cattail, thanks Melissa, and tule. So cattail in purple, tule is kind of in this green patch here where it's very like red when we're looking at the start of season. And then we have those 2 terrestrial water primrose here in this brown, and then water primrose aquatic, where it's just growing over open water here in green. And so when we look at the actual time series of this, we can see that both aquatic and terrestrial primrose have different phenologies. Aquatic primrose, and this is just what I'm referencing it as in this presentation, starts growing earlier and has a longer growing season than terrestrial. And then EMR cattail, emergent vegetation cattail, the start of season is closer to water primrose than EMR tule. So like right here in this box, you can see that their start dates are much closer. And so that might mean that EMR tule is potentially more resistant to invasion because it has a, EMR cattail is more resistant because it has a similar start date to both primrose types. And then because tule has a later date, they then have a chance to start like grow over the tule, whereas they can't do that with the cattail. So water primrose may have a phenological advantage over its competitors, explained by both priority effects and niche breadth. And then it also appears to have an advantage given environmental plasticity because we saw the same results across all different sub-study areas. Then I'm going to quickly highlight some work from one of my collaborators. He did investigate looking at differences between the airborne imaging spectroscopy data from a plane and a drone. And then so this drone would really get you to be able to look at these fine patches where it's starting to encroach on marsh vegetation. And then he also did a cost assessment in his paper. And so that is his paper. I recommend checking it out. This is really important because we can use these mapping to help us figure out what areas are commonly invaded and hopefully help decide when and where to maybe spray or to treat this invasive vegetation. I'm going to just stop my presentation now.
Thank you, Christiana. That was great. Okay, so our next presentation is from Usman Mohammed with University of Florida Indian River Research and Education Center. So please feel free to share what you are. That's great.
Can you all hear me?
Yes, we
Okay.
can hear you, and your slide looks great.
Thank you. Hello, everyone. My name is Usman Mohammed, and I'm a master's student at the University of Florida working on biological control of invasive plant species under the supervision of Dr. Kerry Mintier. And today I'll be sharing with you some of the work we've been doing using drone to assess natural enemy impact in water hyacinth specifically. And if you remember, the first three presenters spoke about water hyacinth, and that's to tell you how prevalent water hyacinth is. Just a bit of introduction. So water hyacinth is really a fast-growing, free-floating aquatic plant that produces these beautiful purple flowers that makes it really attractive for ornamental and garden pool. And we've seen in Dr. Melissa Smith's presentation that this plant is still on sale, unfortunately. So water hyacinth reproduce vegetatively. Water hyacinth reproduce vegetatively, and it forms these dense interconnected mats that impede access to navigation, to recreational and agricultural areas. This plant is native to South America, but has spread globally and is considered one of the world's worst invasive aquatic weed. Here in Florida, this plant is classified as a Category 1 invasive plant by the Florida Invasive Species Council. Category 1 plant are plants that are altering native plant community, displacing native species, or hybridizing with natives. Just like every other plant, there are different control methods used in managing water hyacinth. The first that I have here is mechanical control. This basically involves hand pulling of water hyacinth or the use of mechanical harvesters, as you can see from here. This method is really effective in small water bodies or in areas with small infestation, but can be really time-consuming and very expensive in large water bodies or area with large infestation. Also, there's chemical control, and this involves the use of herbicide to manage water hyacinth. And some great success have been achieved with the use of chemical to manage water hyacinth. However, this method often requires repeated application, and there's a lot of public concern over the use of chemical in waterways. Additionally, the use of chemical can have negative impact on our natural enemy population. And in Florida, there are different biocontrol agents that were released to manage water hyacinth. The first were two Argentine weevils. Those are the early biocontrol that were released in the 70s. And we have Nicotina ecornea and then Nicotina brucei that was released in 1972 and 1974, respectively. Both of these agents are widely distributed and cause substantial damage to water hyacinth. As you can see from here, this is what Nicotina damage looks like. The immature stages of these two species feed internally on water hyacinth, and whenever herbicide are sprayed, it affects their population. And given that limitation, a new agent was released. That is Megamelis cutularis, is a hemipteran, and it was recently released in Florida in 2010. So this is a great agent for IPM, as all stages of this agent feed externally on water hyacinth. So it's more compatible with herbicide. So a great agent for IPM. So what we wanted to do is to assess impact, and you can think like, why are we doing this when we know that the agent are well established? I remember the first presenter mentioned that it's really important to keep like re-evaluating and then monitoring those agents after releasing. So assessing impact is part of biological control. It helps in evaluating efficacy and in identifying areas where agents are underperforming in order to be able to improve the system. However, most of the method used in surveying and monitoring aquatic habitat are traditional method using airboat or helicopters. This method can be really challenging, as remote areas and aquatic habitat are difficult to assess, can be very expensive, and it's also time consuming and very dangerous. And talking about danger, if you're familiar with the habitat, the aquatic habitat in Florida, you know that we have a lot of dangerous reptiles, the alligators. Here is some of the numbers of alligator we have in this small area. And this is how huge these alligators can be. So this is really life-threatening. This is a picture I took from one of our field sites in Lake Okeechobee. So given all this limitation, how do we get into those habitats to be able to get the data we need in order to improve this system? This is where remote sensing comes to play. Remote sensing basically involves the collection of an information from an object without being in physical contact with that object. And both satellite manned aircraft and unmanned aircraft have been used. However, the use of unmanned aircraft provide a safe, quick, and efficient survey. It has better spatial and temporal resolution and has a lower investment when compared to satellite and unmanned aircraft. So the objective of our study was to detect water damage of a 2-by-control agent, and then develop algorithm that can discriminate between the damages. So we conducted this study at the Biological Control Research and Containment Laboratory here in Fort Pierce, using those small tanks, as you can see here. So we randomly selected 3 similarly sized water hyacinth plants, and then we placed them in the tank, and then we added 5 liters of water and some fertilizers. So here's our table of treatment. We have the control treatment with no insect. We have our Megamelis cutularis, we have Nicotina, and then we have the combination of Megamelis and Nicotina. And each of these treatment has 3 different infestation levels, and we have with 6 repetition. So we expose them for 2 and 4 different weeks. And after that, we move on scanning the plant. So the scanning was conducted in Gainesville at the scanning plant facility using a head wall. A head wall has 3 different sensors on it. It has a hyperspectral camera, a thermal camera, and a LiDAR camera. You can see from this picture, this is what the complete structure of the structure looks like. Is an indoor facility, and then here we have the sensors, and then the lighting source that provide lighting to the sensors. So we place our plant in this plastic container, and then we added water just to simulate field condition, and then we proceed on scanning. So we collected our data using Spot, as I mentioned earlier, and here you can see this is a flow chart of all the image processing, from data collection to training and validation. So after collecting our data, we proceeded on applying radiance and reflection correction, and this is done to correct for any sensor noise. And after that, we proceeded on creating reference spectra. And this was done by visually identifying pixels that correspond to healthy plant, meaning the control plant that look healthy, and pixels that correspond to Nicotina damage or megamelus damage. So I did this in MV by visually identifying each of these damages. And then we use this reference spectra in classifying our images using two different classification algorithms, PLSD and SVM, that stand for partially square discriminant analysis and support vector machine. So both are classification algorithms that are used for image classification, modeling, and for predictions. And then we divided our data set into two, and then we used the first data set for training and then the other data set for validation. So here is just visual observation of the plant after the 2 and 4 weeks of exposure. And you can see from here that after these 2 and 4 weeks of exposure, the control plants still appear healthy with kind of few coloration that resulting from senescence. However, our biocontrol plant kind of show moderate to severe damage, especially Nicotina and the combination at medium and high infestation. So you can see those plants are kind of like looking half dead after the 4 weeks of infestation. So for the classification, what we did was we assess accuracy for the 2 weeks data. So remember, we exposed this instead for 2 and 4 different weeks. So we assess accuracy for different weeks separately and then 4 weeks. And later on, we combine the data to assess accuracy for the general model. And you can see that PLSD only achieve high accuracy after the 4 week of exposure, both for training and validation. But then SVM was able to achieve high accuracy both at 2 weeks and then at 4 weeks of infestation. The interesting thing, thank you. The interesting thing here is if you look closely, you see that both algorithms were able to improve in classification from 2 weeks to 4 weeks, meaning that those algorithms are able to learn from pattern and relationship in this data set and then enhance classification. We also assess performance in relation to PCA components. So here what we did was kind of looking at the best principal component that give the highest classification while capturing the most important variance between the training and validation dataset. And from the y-axis, we have the accuracy, and then the x-axis, we have number of PCA component. And if you look at, for example, the PLSD, you see that as principal component increases, both training and validation accuracy kind of like fluctuate. And then once this number increases to 8, we started seeing kind of like the algorithm started stabilizing, meaning that there's kind of like an potential overfitting in this model. So for both algorithm, the 7 principal component was optimum, shows like the best, give us the best training and validation accuracy for a data set. You can see from here, with the PLSD, the 7 component give us the best, and also the same for the SVM. And both of them show kind of like similar trend where accuracy fluctuates with an increase in principal component. So we also employ PLAs and PCA to kind of visualize and analyze patterns in the spectral. And you can see here we have the different classes. Thank you. So we have the healthy plant, which is the control plant. It's kind of like clustered toward one region from this plot, and also the non-insect damage. So something I didn't mention was we also included those damages, like physical damages that were not from biocontrol agents, so that the model will not misclassify those. And you can see here, we're able to mask up that. If you look at this region, we have the non-insect damage clustered together. But for the 2 biocontrol damages, you will see they're kind of like scattered. And this could be because of the different infestation level we have. Because each infestation level will have different kind of like damage. And the same pattern was seen with the SVM model. where the healthy plant were clustered together. The non-insect were also clustered, but our biocontrol were kind of like dispersed all over with kind of like some overlap between Nicotina and Megamelus, which could be very hard at that point for the model to discriminate between those two damages, especially at early infestation. So with this, we conclude that both algorithm were able to classify damages achieving high accuracy, and hyperspectral remote sensing, when coupled with classification algorithm, can classify biocontrol damage and can provide solutions to most of the challenges and the limitation faced with traditional survey method. So the take-home message from here is we already have kind of like a spectral, we created this reference spectra that can be used whenever we fly our drone in the field. We can use this same reference spectra to be able to tell if those biocontrol agents are out there. If not, we can, use drone to do our releases. So this basically come from the idea of the indicative releases in South Africa. So we're trying to see what are the damage levels that we have out there and how to be able to, do some indicative releases too here in Florida. Thank you so much for your attention.
Thank you, Usman. That was very interesting. All right, our last speaker in the post-release monitoring section is Dr. Wesley Daniel with USGS's Wetland and Aquatic Research Center. We are currently seeing a spreadsheet.
Sorry, let me try again.
Okay.
Okay, how about now?
Yep, that looks perfect.
Thank you, appreciate that. Click the wrong button. Thank you for the opportunity to present today on a little bit of our work to increase our ability to do surveillance and track non-native species. Again, I'm Wesley Daniel. I'm with USGS out of the Wetland Aquatic Research Center, and I have the pleasure of coordinating the non-indigenous aquatic species database. And today I'm going to talk a little bit about our efforts to integrate environmental DNA as a new source of information within the database. So if you're not familiar with the non-indigenous aquatic species database, or you'll hear me call it the NAS database, NAS, we are the central repository for non-native and invasive species data for any species that is associated with the aquatic realm, both freshwater and marine. We track just over 1,400 species with all the taxa that are here represented, both plants and animals and invertebrates. And we have just under 720,000 records in the database, which does not make us the largest curation or digital museum of this type of information, but we do stand behind every record, as each record is verified by an expert, both for spatial and taxonomic accuracy. If you're interested in the database, I have a QR code here that you can use to explore. And I would encourage you thinking about signing up for our alert system, which emails out information on any new introduction to the country, to a state, a county, or a drainage, or a particular taxa group of your interest. You get to decide that and opt into which of those alerts you would like. So why are we adding environmental DNA, eDNA, to the NAS database? Well, we have a long-standing relationship with our science and management communities, integrating information on traditional sightings, and that would be from museum samples or photographs. And we really feel like the integration of eDNA helps significantly improve our ability to find those non-native species earlier in the invasion cycle. So really see this as an opportunity to develop a new capacity for early detection and rapid response. And just in case you're not familiar with environmental DNA, it's that genetic material that's sloughed or excreted or otherwise just kind of released into the environment. And I've got lots of examples here from living organisms. And animals are a little better about it because we're a little dirtier, plants less so. And of course, we're focused on that aquatic side, even though eDNA can be extracted from soil and from the air also. So when we started to go down this path of integrating eDNA into the database, we really had a few things or goals in mind. First one is we really needed to establish and maintain some type of data standards. And we wanted these to be community-based, where multiple scientists with their various inferences and their practices all came together and kind of came to a consensus on what those data standards should be. This was important, as traditional sightings coming from photographs or videos or museum vouchers, you always have the ability to go back and verify the accuracy of that identification. eDNA, unfortunately, we don't have that ability. So we really need to be able to trust the methods that are being used, as that making sure they're rigorous enough not to give us any false positives. The other part is we wanted to be able to communicate with our managers, scientists, and stakeholders, and really provide them with this information in a quick enough way for them to be able to respond, but also, again, with those community standards being rigorous enough that we're not making any unnecessary responses. We don't want those false positives. And lastly, we want to provide all this information in a singular location. Before we develop this database, there are a couple other eDNA databases. There's one with the Forest Service of eDNA Atlas, some individual ones from maybe universities or groups. But there really wasn't one that was integrating all forms of eDNA information, and that's what we're doing. We're bringing in information from agencies, from states, from feds, from tribal groups, from academics, all into one singular location. So again, one of the major points about this was we had to come together and make some decisions on what were those standards, because we really wanted to avoid really thinking about those false positives and those false negatives. And so this was a key point in the process. And I could talk 45 minutes long. I can give a full webinar just on how we approach this question, but I'm going to try to sum it up into 2 slides. First one is we developed this 3-step process in how we were going to develop these standards to be integrated, used to integrate data into the database. And so we looked towards the existing literature. We reviewed everything that's being done, not only in the coterminous US and Alaska and Hawaii, but also in Canada. And we thought critically about what were the different field and laboratory process and data analysis steps that are being utilized, and what are those best practices. Then we brought together lots of stakeholders, both within the Department of Interior and other federal stakeholders, along with our broad eDNA community and other practitioners, to really think critically about what those standards are and give us feedback. And then once all of that, and we had consensus, then we developed this web application that you'll see today on the database. So those initial authors are listed here, and I appreciate all their contributions. Jason Ferrante used to be with USGS, and now he works for FDA. But they all contributed in big ways, thinking critically about what are these data standards. And once we had kind of a draft view of what those standards should be, then we kind of went through this iteration process where we went through multiple other reviews. We went further and further out in this little bullseye to the outer rings, bringing in more and more people to review and to comment and to, criticize what we had kind of come up with. So again, we reached out to other government agencies, universities, NGOs. So there was a lot of opportunities for individuals to contribute to this effort. And this is what we came up with. So this is actual data on the non-indigenous aquatic species, or NES database. If you want to get to this page live, you can use the QR code in the upper corner. This is for zebra mussels. So the first thing that was decided through that iteration process is that we would make sure that there wasn't confusion between traditional specimens that you see here in the circles. These are traditional sightings, and eDNA records. And so before you can even see the eDNA, you have to opt into it. So over there in the left bottom corner, you'll see this little image, and it says eDNA records, and you have to select that to be able to see the actual data. And this was an important point for a lot of managers that are maybe uncomfortable with eDNA and are fearful of misinterpretation of what eDNA means. Well, the next step we took was made this disclaimer. we want to make sure that people understand what eDNA means in a kind of concise way. And so when you click that little button to add eDNA to the map, this will pop up and you have to click through it. And we have additional information on the website, provide additional references and guidance about how to interpret eDNA detections. So here is real data within the database. This is in Montana, looking at both positive and negative detections. And one of the great things about eDNA and the stringency of our standards is that we can show negative data, which is wonderful because obviously if we're showing negative data, there's confidence that, that's a spot where we have looked and not found any of the eDNA for the organism, in this case, zebra mussels. So you can see in this example, we have Wolf Lake and the surrounding lakes that do have positive, but that other lake further north, which doesn't show a name on it right now, has that black square showing a non-detect. Now, within each one of those points is a lot of information behind it, much like every other data point within the database. You click on one, you'll get this pop-up showing general, if it's a detect or non-detect. But then if you select an individual here one, so here we selected one of those particular points, then it'll pull up more information. You get the spatial information, you get the day and year and month that it was collected, a little bit about the collection process. This was a water collection. They sampled 3,000 liters. They used qPCR. And they had multiple replicates. There's actually a lot more information behind the scenes, but for our public facing, we figured this was sufficient, again, providing just kind of a concise amount of information that's important. And most importantly, we have a reference there of the individuals and the work they're doing with it that you could always follow up with. And if you're really interested in all this effort and how we came to these standards in the process, we have a publication on that. And again, here's a QR code in the left corner that you could use to get to that paper and hear more about how we made the decisions we did and some of the lessons we learned on the way. Now, if you're interested in providing data to the database, we have a screening process. And if you're interested in the screening page, it's on the right-hand corner with the QR code. The first thing you do, you have to initially make a request for an application. So if you go to the website, there's a couple field boxes you have to fill in, and then we will send you an application link with a unique URL code. And you fill this out online, and this gives us an ability to kind of assess your methods and how you're approaching your science. If approved, then we'll send you a link to a data template and you can fill in all your information. And the great part of this is if you're using kind of a standard set of methods, and we are doing PCR right now. I was going to talk about that at the end, but right now we're just taking PCR data. You won't have to do this every time. This is kind of a one-time thing to kind of verify that the methods meet our standards for data being entered into the database. And once it's done, you're kind of greenlit unless you change methods between maybe taxa groups or locations that you're sampling. Now, I will stress, there are going to be times that people are going to want to submit data to us that don't meet our standards. It does not mean the data is bad. It does not mean the methods are bad. We just had to make some kind of threshold. And the decisions we were made through the consensus building, we're trying to be conservative. Again, we'd rather not have any of those false positives. So we're erring on the side of being a little more conservative to prevent any risk of misinforming managers of potential detections of a non-native species. So this is probably the most important part, and one of the things I definitely want to make sure is well understood. eDNA can be a little controversial, and I know there's managers out there who are good colleagues of mine that are uncomfortable with the idea of having these data points showing up publicly, and that's okay. I appreciate that. So to try to help with that, we've developed a communication process with our jurisdictional authorities. And I say jurisdictional because that could be state, county, tribal, basically anyone who has the legal authority in charge of a non-Indigenous introduction. So through a communication plan that is being developed with the Department of Interior's eDNA communication team, we're going to be able to work with these jurisdictions to make sure everyone has prompt reporting, but we are not getting ahead of a state or a tribe or anyone else with the information. We want to make sure that the proper authorities are informed first before things are made publicly. So through this communication plan that we're developing, we'll be able to plan how data will be released, and there will be the opportunity to embargo data. So if for the first occurrence were to happen in your state, you would obviously want to have an opportunity to respond to that. Maybe additional sampling. It could be making sure you have press releases in order. And so we want to make sure there's room for that for all of our jurisdictions and not to get ahead of them. We're really here to work with you. The goal is not to be the first to get the news out. The goal here is to make sure everyone's informed. And so this data plan, the communication plan, was initially going to be made by my team. But as it's been deemed very important, it's raised the level of the Department of Interior. And so we're making one kind of master communication plan for all these efforts. So that would be the same for Fish and Wildlife or BLM or the Park Service. And so we all would have the same kind of communication plan. So just to kind of sum it up, some of the benefits of this community census was we really were able to kind of create a centralized view of what were our data standards. And again, this isn't like government standard. These are standards for data to occur on the NES database. And by creating this place for eDNA, we really think we're going to be able to benefit the greater community by providing resources that might have been distributed kind of all over in the lit or on individual websites. And now we're able to aggregate it all into one place. And of course, as I said, this right now is for PCR with metabarcoding coming very, very soon. So I want to thank everyone for the opportunity. And this is my wonderful team I always get to work with. I always end with this slide. So thank you very much.
Thank you so much. Very interesting. Okay, so our next and last section is focused on new targets, and this is even more rapid fire than our previous sections. So each one of these speakers is going to be covering probably multiple, sometimes just one, target species, and they each get 10 minutes. So good luck to you all. Our first presenter is Philip Weil with CABI, and I see you are ready to go, Phil, so thank you so much.
Thanks a lot, Melissa. I'm just kidding. So I will be talking to you about some new targets. I do have a very short time to talk to you about 4 different species. So it's really just about creating awareness and updating folks on where we're at with the different projects. This talk was put together by Nate Harms and myself, but I do just want to mention that this is obviously a team effort, and there's a whole bunch of people working in the background on all of these target species. And it does not want to. There we go. So just to reiterate the biocontrol development pipeline, when we're talking about new targets, what we're really talking about is in this bottom end here. So typically in the native range surveys or in quarantine or laboratory cabbies based in the native range. So often we're working in labs, not in quarantine, but essentially down here at this end of the biocontrol development pipeline. And native range surveys may include things like genetic surveys, exploration for herbivores, field observations, and that kind of thing. Whereas the quarantine or laboratory work would include host range or safety studies, impact studies, development of determination of basic biology, and then ultimately submission of the petitions for release. And I can't stress more how long this process can take. It can take anywhere from upwards of 10 years to get an agent from discovery or from those native range surveys all the way through to release. So the first target that I'd like to talk to you about is floating heart or Nymphoides peltata. So this is work that was done by Nate Harms and his team with the Army Corps. The project started in 2019, and they spent a lot of time looking at the genetics. I actually can't see the slide entirely. Looking at the genetics and collecting samples from across the invaded range as well as the native range. And in addition to that, they also did a whole bunch of work on foreign exploration for potential biocontrol agents. And so currently this project is kind of on ice as to where they're at. So they've got a couple of agents, which I'll talk to you about in a second. But the project is currently not ongoing. So in terms of the genetics, what was quite interesting was that they were able to pin down the region of origin of the North American population. And that's really, so if we look at this principal components analysis, which essentially puts different populations in a graph of similarity. And what we see is that the North American populations, these red triangles, come out with the European population, whereas the Asian population really stands out on its own. So what's quite interesting there is that when with the surveys for natural enemies across Europe, very few natural enemies were found, which indicates that the population or that floating hearts is probably introduced both into Europe and into North America. So it's likely that it's native to Asia and then introduced to Europe and across into North America. So although the North American population is most closely related to Europe, majority of the native range surveys were then done in Asia, and those were focusing on South Korea. And a whole suite of agents were discovered, of which two were kind of prioritized as potentially damaging. So this is the Bagus weevil, which is a seed pod feeder, and they consume an incredible number of seeds. And that really has quite a significant impact in terms of seed production. In addition to that, a Puccinia rust was also found in Korea, which does hold potential for a biocontrol agent. Now, during the native range surveys, a Septoria pathogen was also discovered in the US. And Nate may have to correct me if I'm wrong, but this is not necessarily damaging enough to control the populations in North America. And additional biocontrol agents would be required to get the populations under control. Okay, the next target that I'd like to bring attention to is parrot's feather or Myrifilum aquaticum. And there were a couple of questions on how the agents may do in cooler climates. So we're actually working for Canada and sort of the northwestern part of North America, so Canada and Washington. And we're very interested in that, and we're currently working on the thermophysiology of the agents. So we are in the quarantine or laboratory phase here, where we're looking at host range studies of two agents which we currently have in the lab, Lysathia and Phytobius festitus. And then Listrinotus we have in our other cabby facility in the UK, and we're starting to look at the host range testing of that as well. And this project started in 2021. Okay, the third target that I'd like to bring your attention to is a European frogbit or Hydrocharis morsus ranae. So this project started in 2024, so just this year, and it's funded by the Army Corps of Engineers and led by Ian Knight over there. And we're really in the native range surveys, sort of in the pipeline at the moment, where we're looking at collecting genetic samples across both the native and the invaded range to try and pin down that region of origin. And that'll really help guide us where to search for potential biocontrol agents. In addition to that, we were able to do quite a considerable amount of surveys this year, and we've already found two of our prioritized potential biocontrol agents, Hydrelia albifrons, which is a leaf mining fly. You can see the larval mines in the top right-hand picture here. It appears to be quite a damaging fly, taking out that kind of surface area for photosynthesis, and also structurally damaging the plant and kind of sinking those leaves. It's found virtually at every site that we've surveyed. It can have multiple generations, and we do really feel like this species could hold potential. Often Hydrelia flies are quite specific, so we're quite positive in terms of safety as well. In addition to the fly, we did find one Bagus species, which we suspect is Bagus puncticolus, which is, we only found it at one site in Hungary, and we were unable to get any eggs, so we haven't made much progress in terms of the lab work. But we do plan next year to try and source different or source additional sites where we can collect more weevils. Now, coupled with this project, we've also been able to start some work on water soldier. Water soldier is only found in Canada at the moment, but it is spreading quite rapidly, and we suspect that it will make it into the US in time. It's already found. It is already sort of making its way down. So this is the surveys are largely coupled with the hydrochorus project, mainly because of their phylogenetic relationship and the fact that the sites generally overlap. So the plants are often found at the same sites or in the same region. So again, we're in this sort of native range survey area where we're collecting genetic samples across the populations to try and pin down that region of origin, and also into the foreign exploration for potential biocontrol agents. And ironically, we're in a very similar situation as European frogbit, where we have one Hydrelia species, Hydrelia tarsata. This is a larvae here inside the leaf, and you can see the mine. This is also found at several of the sites that we've surveyed, and does also appear to be damaging, and again has the same positive aspect that majority of these species are fairly specific. We've also found one weevil species, Bagus binodulus, and although it's quite, we found a fairly large population of this weevil, we've also only recovered it at one of the sites. And with that, I would like to thank you very much for your attention. My email address is up there. If you do have any questions or would like more information on the projects, please feel free to reach out. And I'd also like to thank all of our sponsors, collaborators, friends, and colleagues. Thank you very much.
Thanks, Bill. All right, our next speaker is Wade Simmons with Cornell University.
Hey, good afternoon, everyone. My name is Wade Simmons. I've been working on the water chestnut biocontrol system here at Cornell University for the past few years. And in my presentation today, I want to give an overview of some of our big research findings and provide an update on the next steps for this program. Okay, so water chestnut is a pretty unique aquatic plant in that it's an annual species that both roots in the sediment and produces these rosettes of floating leaves that come up to the water surface. Like many of the plants talked about today, it's got a pretty explosive growth rate. It has a high reproductive output, and it also has this habitat preference of shallow, slow-moving, and nutrient-rich fresh waters. Now, water chestnut can become particularly problematic when it forms these large, extensive mats that can transform these open water, freshwater systems. And this physical modification can drive large changes to both the chemistry of the system, mostly through depletions of dissolved oxygen, and have big implications for both ecosystem processes like nutrient cycling, but also organisms that are sensitive to changes in these oxygen dynamics. So the insect that we've been working with here at the quarantine lab at Cornell is Galaricella burmanica, or the water chestnut beetle. This is a leaf-feeding beetle whose both adult and larval forms feed directly on the leaves and photosynthetic material of the plant. There are multiple generations of this insect per year overlapping, and the females have a high reproductive output. So this is very hopeful for large, rapid population growth if this insect were to ever be approved for release. Now, some of you might already be familiar with some Galaricella insects. There were two released in the 1990s into North America for the biological control of purple loosestrife. And even though they don't really appear similar, both purple loosestrife and water chestnut are both highly related. They're both in this plant family Lythraceae. And there's something about the shared chemistry of this Lythraceae group of plants that Galericellas have developed associations for. So here it's kind of a new pairing, but a familiar pairing between a Galericella and a loosestrife member. So I am happy to announce that we have completed the research phase of the biocontrol research for Galericella bermanica and water chestnut. And we are now actively preparing the tag petition for regulatory review, which we hope to submit here in the coming months. Our research over the past few years has focused on these three key themes. One is the effectiveness of how much will this biocontrol suppress water chestnut populations? And then two aspects of safety. What is the host specificity of this insect we're hoping to release? And what are also some of these possible new interactions with related co-occurring species? Okay, so let me just walk through some of the big highlights from those research themes. So this appears as though it's going to be a highly damaging and effective control on water chestnut populations. We used studies from 5 populations of water chestnut here in North America to inform these population models, and we took impact data from the native range to estimate what the changes in population trajectory would be over time under different scenarios of biological control. And even taking a very conservative estimate of 30 to 40% reduction in seed set under biocontrol, what we see is a shrinking population of water chestnut populations over time, which is exactly what we're hoping to achieve with an effective biocontrol program. We've also tested the preference and the performance of Galericella bermanica on multiple populations of water chestnut that occur here in North America. When we started, these were all classified still as Traponatans, but now one has been recently reclassified as Trapa bispinosa, or rather identified as Trapa bispinosa, which is co-occurring in Virginia and Maryland. The data is great in suggesting that these insects can attack and develop on a wide range of these populations. And there's some indication from our data that there's even a slight preference for this new arrival of Trapa bispinosa, although we need to design a more formal experiment to piece that apart. But the good thing is that all these populations look vulnerable for biological control damage. So how about the safety of the insect that we're looking at? This is just a representation of the host specificity testing process that we've undergone. And in the first level, we've exposed 57 plant species here in North America to the insect in starvation, simple reductionist Petri dish type of studies. And of those 57, only 9 had any feeding damage or eggs laid upon them, advancing to this more ecologically realistic testing arena in multiple choice testing. Now, this is what this multiple choice setup looked like. These were these enclosed ponds in which we had an array of water chestnut, of these species here. And we came back after 5 days after letting them be to count damage and eggs. And what we're looking at here is a majority of green, which is suggesting that our evidence is highly attuned to the water chestnut. All the eggs and most of the eggs and damage were on water chestnut. However, you'll see this orange color in here too. This is the native plant, Brasinia schreberi, which we have confirmed that our insect can also develop on, although limited development. So this echoes evidence in China that shows that the insect can use this plant. However, it fails to establish big populations. You can see in these graphs, these 2 different lines. Here at the bottom is the insect feeding only on this non-native or non-target host Brasinia, which it fails to establish populations. So we also have some other evidence here to suggest that these impacts to the native plant here will not be super impactful. So in Asia, we have no evidence of this herbivory driving declines in Brasinia plant populations. And we also have native data here that This plant is widely attacked by native insects, yet populations appear robust. So while we do expect some minor feeding damage on this native plant, we do not expect any of this to cross a threshold to affecting population levels of this plant, which is really a safeguard for the plant. Now, adding some complication to all of this is that we are aiming to introduce this insect to water chestnut beds that already have a native insect occurring on them. And so we've spent a lot of time over the past few years trying to explore these interactions between the insect we want to introduce and the resident native insect already occurring. Now, a fundamental question we wanted to answer is, could these insects potentially hybridize? And if so, what do those hybrids do relative to the two parental species here? Now, we've done a series of tests here, but the main takeaway is that yes, some amount of hybridization is possible. We've been able to force mate these two species in the lab to produce viable offsprings. And we've gone through a series of tests here that look at comparison of performance and preference of these hybrids relative to the biocontrol and the native. Now, there's some reassuring evidence from these hybrid trials. The mating between species is uncommon, but can be forced. So it's biologically possible, but ecologically, we're unsure. There's no signs of improved performance of these hybrids, and eggs and feeding on the 3 expected species only. So I see I'm quickly out of time here, but I want to just point out that these hybrids are kind of a predictable intermediary between these two species. And the evidence that we have does not suggest that these impacts to the native species, Brasinia or Nufar, will account to any kind of population level impact. So in conclusion, we're done with the research phase here and we're putting this all together. We have very promising effectiveness. We have risks to native plants appearing low, and we have this kind of uncertainty with hybridization that is biologically possible, but the evidence suggests that this might not be ecologically realistic, and even if so, this should not amount to population level impacts on plants. Thank you very much.
Thank you, Wade. All right, our next and final speaker of the day is Dr. Flaminia Mariani with Louisiana State University.
Good morning, everybody. Can you see me?
Yes. Thank you so much.
Good morning, everyone. My name is Flaminia Mariani, and I'm a postdoctoral researcher at Louisiana State University. Today, I want to talk about part of my PhD research, which I carried out in Italy. The topic of the research is biological control of duckweeds. And in particular, today, I would like to talk about one species of duckweed, Lemna minuta. This plant is native to North America, and it is one of the smallest flowering plants in the world. Every plant, as you can see from this picture, is measured only 1 or 2 millimeters. This plant has arrived in Europe in the 40s and in Italy in the 90s, where it has become one of the alien species with the highest potential for invasiveness. This is due to its high vegetative growth rate. It is estimated that in 2 or 3 plants, in optimal conditions, it can double its biomass. At first, this plant colonized the entire surface of the water body, and then it started to grow in thickness, creating floating multi-layer mats on the surface of the water body that sometimes can reach a thickness of even 10 centimeters. And this causes alteration in the water body. In particular, I want to give you a quick overview of the impact of the species in the invaded aquatic sites. And to do that, I want to talk briefly about the research that I carried out at the beginning of my PhD in Italy in 70 pairs of sites. For every pair, the 2 sites were very similar from a microenvironmental point of view, but they differ for the presence or the absence of this plant. What we found from this study is that some factor, as the light in the water or the dissolved oxygen, they were negatively correlated with the increased coverage and thickness of the lamina minuta mats. And this had effects on the plant community, of course, because in the sites with the mat of duckweed, the number of species was lower and the overall plant coverage was lower, with the disappearance of some important key taxa of rooted plants and of macroalgae. And the same happened for the animal community. Indeed, it was found that in sites with duckweed mats, they had a lower richness of species and the communities were altered in their specific composition, with the prevalence of species that can tolerate anoxic conditions, while the species that were more sensitive to lack of oxygen, they were absent or rare. And in the same way, also the insects that have a winged adult phase or that swim on the surface of the water. And this study was published, and I'm leaving here for you a QR code that you can scan if you want more details about this study. So given these very strong impacts, managing the species is important. And herbicides are effective, but the ones that works better on duckweeds, they were banned in Italy, so we couldn't use them. Mechanical removal has a lower impact on the ecosystem, and we try to use it, but it has proven not to be effective on a plant that is so small and fast-growing as lamina minuta. So because of these challenges, I was very interested in biological control of this species, and in particular in classical biological control that is based on the use of a non-native organism as a biocontrol agent. We have just seen in the previous presentation how effective this has been for some of the floric invasive plants that we talk about. But I was doing my PhD in Italy, where there is still a lot of skepticism about this method. Indeed, the scientists that were in my PhD committee, they were mostly conservation biology, ecologists, and botanists. They did not approve this method because they were worried about the risks of introducing a non-native species as a control agent. So I decided to try a different strategy, using native natural enemies. And I found a native aquatic moth, which normally feeds on native duckweeds, that turned out to feed on the alien duckweed, with an interaction that fits with the new association hypothesis. This type of approach was really well received by the academic community I was in, and I got a lot of very positive feedback. But I was still very interested in classical biological control. So I decided to write a project and apply for a Fulbright scholarship that would give me the opportunity eventually to travel to the native range of lamina minuta and look for its natural enemies in the native range, since there was still no information about that. So I won that scholarship, and in the summer of 2021, I was able to carry out service in 68 sites populated by duckweeds in 2 states of the United States, in California, that has a Mediterranean climate similar to Italy, and in Louisiana, which is rich, very rich in wetlands that host a high diversity of duckweeds. And we discovered 3 herbivore insects associated with duckweeds belonging to the orders of Coleoptera, Lepidoptera, and Diptera. These findings were significant because this was the first survey targeting natural enemies of lamina minuta in the native range. But after reviewing the literature, I found out that the host range of the Lepidoptera was too broad. So I ruled out this option and focused the rest of my studies on the fly and on the weevil. And with morphological and genetical analysis, we confirmed the identification of the 2 species. The weevil is Tanispirus lemne, and the fly is Lemnophila scotlandi. So I decided to carry out some preliminary host range studies to evaluate the specificity of these 2 insects against lamina minuta. For this experiment, we use 5 duckweed species, including, of course, the ones that are native to Italy and Europe, which are lamina minor and Spirodela bolyrrhiza. The setup of these experiments was a small plastic container filled with water and a monolayer of plants. We made sure that the plants were not overlapping each other. And then all the plants were exposed to 6 adult insects of either the fly or the weevil for 96 hours, after which the insects were removed. For flies, we were able to count the number of eggs. As you can see in the picture, they were very easy to see because they have an elliptical shape and they are laid on the surface of the plant. But for the weevil, the situation was different because the weevil lays the eggs inside the mesophyll of the plant. So instead of counting the eggs, I decided to count the number of the newborn larvae. What we found from this study is that the weevil successfully fed on and laid eggs on all the duckweed species that we were testing. And there were no difference in the number of newborn larvae or on the survivorship of the adults exposed to the plants. So we decided that this experiment showed that the weevil was not specific enough to be considered for the biological control of this plant in Europe. But the situation for the fly was different because the number of eggs that the fly laid on Lemna minor and Lemna minuta was higher than the other native species in Europe, Spirodella. And also the adults exposed to the 2 other genera, Spirodella and Landoltia, they did not survive because they did not feed on these plants in the 96 hours. So these experiments show that Lemnaphila scotlandii could have some specificity at genus level, but this genus level also includes the native to Italy Lemna minor. So for this reason, I decided to carry out a second series of experiments to test if the Lemnaphila scotlandii had any preference between these 2 species, Lemna minor and Lemna minuta. And in this experiment, we test the feeding and pupation preference of both fly and larvae under both choice and no choice condition. The results of this study show that there were no difference in the percentage of damaged plants by either fly or larvae in choice and non-choice conditions. Anyways, there was a preference in the pupation because larvae were pupating more often in Lemna minor, the native plant, than on Lemna minuta, when both the species were available at the same time. And this could probably be due, as you can see in this picture, from the larger to the larger size of Lemna minor that gives more protection to the pupae inside the plant. Also in this case, the study has been published, so I'm leaving a QR code if you want to have more details. So in conclusion, none of the insects that we found in the native range, they were not suitable for due to their broad host range. But the fly has some specificity to genus level. So it could be suitable where other species of Lemna are invasive, but there are no native duckweeds within the same genus. And also more native range surveys are needed to identify more specific agents for Lemna minuta in Europe. Thanks for the attention.
Thank you so much. I love that picture. All right, so now we will have a brief question and answer period before some closing statements. So Jen, do you have some questions that you would like to ask?
Yes, thanks, Melissa. We've got quite a few questions in the question box. We'll start by asking a question for Usman. Did you say you were actively able to drone drop biocontrols in the lower density areas, or would this be a future option?
Okay, so we haven't used drone to release megamelis yet, but it's what we're looking at doing in the future. But if you're talking about like drone releases in general, yes, there have been some release of biocontrol agent on the old world clamping farm using drones. So yes, but for water hyacinth, that's what we are looking at doing in the future. I hope that answered the question.
Great, thank you, Ethan. Our next question is for Phil. For European frogspit and water soldier, does this development process also apply across the border in Canada, or does that need to be done in its own development process?
Yeah, I think this was Matt Boulding. Yes, no, well, yes and no. So the development that process is something generic that happens in all countries or in all regions. But what we do need from each country is a representative who would or a champion who would kind of drive that project forward. So for example, as CABI being an international organization based in Switzerland, we cannot be the ones to submit a petition for release of an agent. That has to come from within Canada or within the US. So for the European Froglet project, we are desperate to get a Canadian representative who would be willing to champion the project, who would be willing to submit the petition when we are ready for that in a few years' time. And to join the consortium. So if you have anybody in mind, please reach out. I know you have my email, and everybody else should hopefully have my email as well. Otherwise, check the recording when that comes out. Thanks.
Thanks so much, Phil. Our next question is from Chris for Wes. How do you deal with missing data in eDNA? There must be a large number of unknowns given the large number of unnamed and even larger number of unsequenced organisms.
Great question. I appreciate that, Chris. So with the qPCR process, you are developing individual species assays, which are targeted for that individual species. That takes quite a bit of time because you do have to validate it, not just for the species, but all of its congeners and other species within the drainage to make sure you don't get any false positives. And so we're not to the point yet where you could just take a vial of water and identify all taxa within it. The metabarcoding process is getting to that point. That is one of the goals, is to be able to run multiple species eDNA assays all together at one time. That's what metabarcoding does. But we're not there yet. So yes, it's still a problem. You have to kind of still think individually about the species of focus. And that's why it can be a good tool for early detection as long as you know what you're looking for.
Great. And Wes, while we have you, I know Joey asked in the post chat if you could explain how your work just briefly relates to biocontrol.
Great question. I did post a response, but yeah, it's about surveillance and monitoring. So eDNA does not directly relate to biocontrol in the sense that it's not a control agent or a process, but you can use it for monitoring, especially when you're starting to think post-control techniques. When you think you have maybe achieved eradication, eDNA can help in that long-term monitoring to make sure that you don't have those small insipid populations still around. So really a tool of early detection, but it can be used for monitoring and other uses associated with biocontrol.
Fantastic. Thank you. And I believe, Wade, you have been answering questions about Galaricella on loosestrife and water chestnut in the chat. So I think I'll jump to the question for Christiana. Is given the cost and time of perfecting your approach for your specific location and your species of interest, I'm wondering what your thoughts are regarding its use in situations in the future. Do you foresee an approach being readily adaptable to other similar systems?
Were you talking about the satellite data or the drone that I mentioned briefly at the end?
I believe it was the satellite data, but
Oh, okay. So the satellite data that I used is free and open access. So in terms of costs associated with it, that would be just like your compute cost, which in some cases might not be that high because some of this can be replicated on Google Earth Engine, which is a free resource. So I do think that this is applicable to other areas, and similar algorithms would be able to distinguish different floating types in other regions. The biggest area of concern would be at the satellite pixel size of 10 meters. You would need to ensure that the patches of your vegetation are at least double that size in order to detect them.
Great, thank you. I think that's all we have time for with questions. And I'll hand it back to you, Melissa.
Great, thank you. So just real quickly, before I turn it back over to Christy to close us out, I just want to say on behalf of the NAISMA Classical Biocontrol Committee, thank you to all of our speakers for taking the time to share your very interesting work with us. And also a big thank you to the USDA Forest Service for sponsoring this and making it free and open to all. And lastly, thank you so much to Jody Hogan with NAISMA for making sure everything ran so smoothly. All right, take it away, Christy.
Thanks, Melissa. And I will echo you as well. We really appreciate all of your efforts, Melissa, Jen, and the rest of our speakers today. Please share your final reactions and help us to thank all those who took time to be with us. In the chat, you'll find a link to the post-event survey. We ask you to take a moment to fill this out. These surveys help us to continue to deliver trainings that support areas of interest that are important to you. A recording of this summit will be posted for you to view in the coming week at naisma.org. A friendly reminder that NAISMA's next and final webinar of 2024 will be focused on Canadian biocontrol efforts, and I encourage you all to join us on December 18th to learn more. Finally, I would like to invite all of you to our upcoming National Forum on Biological Control event, which is organized by many members of the NAISMA Classical Biocontrol Committee, our partners at the National Association of State Foresters, and our generous funders and partners at the USDA Forest Service. This event will be held in Annapolis, Maryland in April of 2025. And as we sign off, a final thank you again to our generous sponsors, USDA Forest Service, for supporting today's events, all of our speakers, and all of you for helping to support NAISMA. Thank you for joining us today and hope to see you all soon. Take care. Goodbye.